Hello, ma'am. We'll start accepting attendees po from Zoom.
So good afternoon. Good afternoon, everyone. So I think ika labing apat o kanan to? 13 talk. So, lucky 13. So, tika labing tatlong talk na si Ma'am Lil in our series ng MSI anniversary. No? So, hindi lang siya ang magsasalita. Tama? So, I heard marami ka pa magsasalita. Pasensya na, hindi ako makastay sa talk nyo. I leave for cruise tomorrow. So, ang daming hinahabol na papeles before that. Pero kilala niyo ba boss nyo? So, si Lil, so she earned her PhD in pharmaceutical science and medicinal chemistry sa UF, University of Florida. At isa siya sa labing-isang bumalik o ngayon labing-tatlo na nagbalik scientist sa MSI. So out of that 13, pito silang naligawan namin na magstay. So si Ma'am Lil yung sa biotech, no? So alam niyo naman yung ginagawa niya sa lab. So she leads, her laboratory leads efforts in purification, structure elucidation, and bioactivity assessment of natural products, using both traditional methods and the advanced methodologies of omics, no? Genomics, metabolomics, and high throughput screening. So marami na kayong na-explore sa marine fauna and flora and fauna. So may bacteria, cyanobacteria, sponges, mollusks. At hindi yung ginagawa ang isa sa magandang ginagawa ng lab nyo is you're just not just doing the isolation and research for yourself, you're also biobanking it so that other researchers going forward, hindi na nila kailangan mag-collect from the wild. They can use the products that you have to further the research. No? So, yun. Hindi nyo alam kay Ma'am Lil, nagkagulatan kami. Dinalaw ako ng kapatid ko dito. My brother is in theater. At nagkabanggaan sila ni Ma'am Lil sa corridor, magkaklase sila pala sila mula elementary, high school, mula kinder. So, hindi nila alam. Nagulat silang dalawa. Yun. So matagal na pala connection naman ni Ma'am Lil noon pa. Nung nagwawala pa kapatid ko, andun na siya. So, thank you po sa patuloy na pagsubaybay sa MSI Talk at 50, no, celebrating our 50th anniversary. So, battle cry natin beyond borders, and I can see si Ma'am Lil, marami ng tao na natulungan to go beyond borders kahit may iyakan. And so, Ma'am Lil, ikaw muna. Tapos ikaw na lang mag-introduce sa iba.
Thanks, Ma'am Lau, for the introduction. Good afternoon, everyone, and thank you for coming today. This is a special lecture series because, as Ma'am Lau said, I'm not the only one speaking today, but I'm also invited my lab to also do the presentation and share our work. And this is in celebration of the 10th year of the Marine Pharmacognosy Lab. So MSI is celebrating 50 years. Our lab is relatively young, celebrating our 10th year anniversary. And it has been a wonderful ride. Some tears, but generally, I think it's a very much wonderful time here at MSI. So for today, I will discuss something old, something new, and something new. That's in TikTok terms. But if you want the longer title, it would be Old Chemical Tricks of Marine Organisms for New Innovations in Today's Blue Economy. And the caricature here indicates or gives you an idea of what are the source organisms that we work on. And this would be from the very simple and to the more complex organisms. Okay? So, Okay, so I would like to think my lab, the overall theme of the lab is marine natural products for Filipino health. Marine natural products is both good and the bad and the ugly. Good because we can find innovations from these marine natural products for industrial and biomedical application. The bad because again, they can be toxins and venoms that can be deadly to humans. So half of the lab would be on biomedical um, research, developing innovations from marine organisms, and the other half would be more or less looking at your harmful algal bloom and their toxins. Okay, so when you go to coral reefs, when you go snorkeling, diving, you feel very much relaxed. Agree? Can I see hands? Yes. Actually, coral reef is a very stressful environment. Ask your marine organisms, and they will tell you 24/7 time, but you have to defend yourself. You have to defend yourself from your competitors for space, for resources. You have to find a way out of predators. Okay, you may be slow or sluggish, or even you cannot move from the seafloor, but yet you have to defend yourself from being eaten. And at the same time, if you're a predator, you also need to find food. So you have to defend yourself from hunger. Right? And then, of course, there's a more special relationships from, say, for example, your corals or something, where you have very intricate relationship, where you have symbiotic relationship. So it's 24/7 trying to put yourself in the coral reef environment, and the rest of this would be mediated by your chemical warfare. So molecules are produced by marine organisms for very specialized function. If they don't produce this, they would just suddenly survive. They would have a much more difficult time. So the natural products, the molecules produced by the marine organisms, there's the competitive advantage, aka growth factor, right? So this relationships and natural products produced by your marine organisms have been validated to be a source of new drugs. An example of which be the Venabuffinus magnus, which was discovered by Vladimir Oliver and first our national scientist Lilibeth Cruz, and this gave rise to the drug Prialt, now marketed and used in clinic. So when we study the production of natural products, we look at several methods. So right now it's not just all about your purifying and doing the structural elucidation, but we also have to establish ourselves and what are the molecules that could potentially be used or produced by your organisms. Okay. And you now have your techniques that use all the way to metabolics. So this would transcend genes to phenotypes. And you know what? Will tell you what can happen. Molecules that would produce research solutions, and from this you need to get your DNA and eventually your whole gene sequencing. And first, we've been very fortunate to have whole genome sequencing done with the group of Lilibeth, and eventually we can also do transcriptomics, and this is very much useful for elucidating the products or the products that are ribosomal included. And then proteomics will tell you what is happening. This would be balancing the machinery, would be the probably the manufacturers of your nutrients and booster proteins. And if you have proteomics in natural products, this actually We got the field for once a year. And sometimes we don't even have to go to the field with our samples because we ask from collaborators if we already have um, existing cultures. So we're hydrophobic in that case. And you see why we need to stay on the left and right? Because after the collection trip, which is step one, then we need to do other with other activities down the road. No? And after the teaching of organisms, whether it's microorganisms, we have to do a high throughput screening and develop the on your application, your desired application. Okay? And after we do a high throughput screening, then we would have identified the extracts with the desired negativity, and subsequently we now do the identification of the slates and then large scale culture and extraction. Okay. Very good. After this, after we already identified the types, then we do structuralization using mass spectrometry, nuclear magnetic resonance spectroscopy, derivatization, for sending radioactive photons. This would be that I will be sharing in my work together with my group on the identification of radioactive compounds with could be new structures. At the same time, we also interface the genome analysis for the identification of student with features. We got to have an idea of how much of the molecules may be produced to be possibly new compounds. Okay? So you now understand why you don't do collections at least once a year, because if we have too much, then we also lose some quality material because we have to finish especially the first part, and would be now going after time and at the same time making most of the resources that we. So since we are in the Philippines, everything is more fun. Shipments included. Shipments made by the economy. We call them commute. And if you've been to Lawan or Flat, you've probably been offered the Ibon Valleys. This would be the British dish that's offered when you go to a mangrove park. And that actually not works, but the mollusk. And Ibon Valleys is one of the most that are classified as your classical shipper, right? And pretty much distributed worldwide. They're also, believe it or not, considered as the food of the future because they grow faster than your clams and oysters, and they're also very protein rich. And with PNS ICBG project, another good buried shipper was identified from Madonna Tigers, and this was the locust um, production of energy from hydrogen sulfide. So shippers, they're very nice system to study your symbiosis. So for your typical shipper that's boring, they would have the sequence the same as digestion. But the sequence is completely devoid of any bacteria. But they would have the cellulosic or carbohydrate degrading material, I'm sorry, in the sequence, and the bacteria will be found in the guts of the shippers. And this is where your enzyme production would occur. And there will be a shipping of the cellular enzyme from the girls to the sequence. So why not have bacteria in the sequence? And that's to maximize the nutrition coming from your carbohydrate degradation. So the cecum is largely devoid of bacteria, and as you saw in the previous example, different shipworm, different lifestyle, different microorganisms. Okay? And for your football shipworm, the quaternary is the major bacteria present in the of your shipworm. And quaternary devotes 7% of the genome to natural splices and this is comparable to your antibiotic producing streptomyces silicosis. So there's a lot of biosynthetic potential for, from your lineage or and so if we look at the microbial symbiote from your shipper, we can see that we have symbiotic relationship with the host, providing the host with carbohydrate degrading enzymes as well as the aid in nutritional fixation and they utilize enzymes to do this. At the same time, the microbial symbionts residing in the shipper would have an antagonistic relationship with microbial pathogens. They don't want the host to be overpowered by your KA bad bacteria. And they utilize this by producing small molecule compounds. And your microbial small molecule compounds, as well as enzymes, can be the subject of biotech and biomedical applications. And at the same time, this can be utilized, or this is can be utilized to mine now your the different compounds that will be produced by your shipworm microbial symbionts. Okay. So during the EIDL project, which was led by myself and together with collaborators from the Institute of Chemistry, Dr. Lilibeth, we undertook analysis on the different strains of the nectar ternary, factor leads and compared this with a strain ternary T791. So this microbial symbiote and two three strains are all coming from the Philippines. Okay? And so the fermenter culture of the ternary and then has subjected this to mass spectrometric analysis. So under mass spectrometric analysis, compare the different molecules produced, and you can see here that the different strains of ternary actually prolific producers of lipids, and also lipids, as well as your the compounds such as your triplets, thermocytomycins, stabilizers, have been to declare the cures produced by the sperms, stimulants, in a while. So let's see here that the microbial stimulants from sperms produce a lot of molecules. Okay. characterize this? No, because much of the work in assessing the BGC of your shipworms are actually based on genomics and metagenomics data. So we collaborated with data from genomics established by PM's ICPG, and at the same time confirmed that different strains of ternary actually have also different capability to produce different compounds. As you can see here, for example, in the mutant viewers or mutant clusters, each bacterial molecule, you can see that where the molecules produced by the strain 2X, it's a lot more compared to the carrier strain E791. Okay, so what are the molecules produced by shippers? Comes in all forms and shapes. You have the anti-apoplectin compounds to have siderophores such as your ternobactin, teridinobactin, and also antibiotics such as your Turner cyclamycins and the shipworm microbial symbionts really want to ensure that no harmful bacteria would persist. So aside from producing your antibiotics, they also produce antibiofilm compounds. So for microbial pathogens that may be difficult to kill because they form biofilms, well, the microorganisms already thought of a plan or devised a plan to do that, and that's to produce biofilm compounds, and we named this ternuric acid, and this was identified by my grad student Noel Lacerna, and subsequently as well, we identified several other molecules such as lumicrome and the hydroxyacetyl indole. So the lumicrome are cytotoxic. So there's some form of killing mechanism that's initiated by the compounds from T. ternary. Lumicrome is a growth stimulator, and our hypothesis would be that it may be involved in the growth of your shipworm larvae. Okay. So looking more closely at the molecules belonging to the pyridinobactin cluster from T. ternary strain 1022X, and this was purified and done by Jeremiah Batukan as part of his MS thesis here at NSI. We show that apart from the described known molecules, pyridinobactin A, and its dichloro analog, strain 1022X also produce a lot of other pyridinobactin analogs. So it seems this strain of 1022X is a particular super producer of your pyridinobactin compounds. Okay, so the first described pyridinobactin was pyridinobactin A, and you can see it in the main group. And apart from that, we also describe a bromo analog of pyridinobactin A, as well as your benzol analog of your pyridinobactins. And there's also a shorter analog of your pyridinobactins, which we call your truncated pyridinobactin A. We're currently doing the synthesis of the truncated group with a group of Eric Schmidt in the University of Utah, and we hope to share with you the results of this later on. So, why produce your pyridinobactins? When Bailey Miller described the bioactivity of pyridinobactin, they showed that it's a siderophore, right? But looking closely at the structure of pyridinobactin A, it actually resembles a new reactive compound known as polycatin. And the polycatins were also purified from a microorganism that's associated with cone snail. So if you look at the structure, you see that pyridinobactin A is just a unit longer compared to polycatin 3. So polycatin 3 is a known neuroactive compound that targets serotonin receptors, and so if they are structurally related, then their bioactivity would be the same. So we tested pyridinobactin A and polycatin within your dorsal root ganglion assay. And what you have to look here would be the change in the fluorimetric ratio. So the y-axis would be the fluorimetric ratio and would indicate the change in calcium influx in your dorsal root ganglion cells. And this would be represented by these traces. So the compounds are treated here, that's 1 micromolar, and subsequently treated at 10 micromolar. And the initial ones would be your baseline level. So what happens to the baseline, to the calcium concentration after the treatment of your sample? So let's look first at pyridinobactin A. So we see we add a KCl pulse, so pulse, and then you put in your sample, and what do you observe with the signal? Is it an increase, decrease, or no change? Increase, thank you, with the 10 micromolar. And then subsequently, you do a baseline pulse, it returns to the same as your initial condition. So this one. So you say it's reversible. The compound is reversible, but it caused an amplification. And then you treat again with your 10 micromolar of pyridinobactin A. What happens with the signal? Increase, decrease, no change. Increase, right? So then your pyridinobactin A amplified your the calcium signaling in your DRG neurons. And if we compare it with polycatin 3, again, you see the same, you do the same sequence. You pulse with KCl, pulse, compound. What happens now? Change, increase, decrease, no change. So baseline, add, there's a slight increase with your polycatin 3. So your pyridinobactin A affected a lot more of the neuronal population, that's about 47%, compared to your polycatin 3, which affected only about 26% of the neuronal population. So what to do now? So we can do a combinatorial treatment and see if we see an additive, a negative, or a negative effect. So again, you do the same thing. You do a pulse with KCl, add your sample. What do you see? You now have an increase, and then treat with your polycatin 3, and you see a further increase in your signal. And so the cells amplified by pyridinobactin A were also amplified by polycatin 3, and we think that polycatin 3 and pyridinobactin A would share the same target in your cells, and because of the similar phenotype for your that's observed in your DRG. But why produce pyridinobactin A? Well, polycatin 3 is a serotonin receptor agonist. Okay, and so our hypothesis now would be pyridinobactin A may be important for the settlement of your shipworm larvae. So again, another molecule, yet another function that it aids your shipworm host. Okay. So coming soon in 2025, we have an NRCP-funded project entitled Tamilok, and we'll look at the shipworm host, microbial symbionts, and do a more systematic analysis of the biodiversity of the shipworm host together with the microbial symbionts. And this will be led by all-power female scientists from MSI, IC, and the Institute of Biology. So that's coming in 2025. We'll be recruiting grad students for this project. So we'll keep you posted with that. Okay? So your shipworms is really a good system to study your symbiotic relationship between the host and also your the microorganisms. And we know that another good study organism would be your sponges. Okay? There has been a lot of interest in this blue sponge. And if you go diving in Puerto Galera, you probably would have seen this blue sponge. And this blue sponge is known to produce cytotoxic molecules called rheniaromycins, and they are related to the already marketed drug ET743. Okay? Now, what's the difficulty with this molecule? Well, if you look at the structure, it looks like it's a problem, right? For the production. Synthesis-wise, it's difficult because you have 5 stereocenters. And when you do your synthesis, you have to get stereochemistry of the chiral centers correct. Okay? And so there's only a limited synthetic route available for your rheniaromycins. And being in a developing country, synthesis is more or less a little bit more difficult compared to doing a straight up purification from the sponge biomass. But then these compounds have a great potential, principally aside from being anti-cancer compounds on their own, they are also molecules that act synergistically with doxorubicin. So you want to design combinatorial regimens of drugs to limit your side effects, right? To limit the formation of having resistance, drug resistance. And so with Jordan Toon and of course Dr. Giselle Concepcion, we worked the combinatorial studies of rheniaromycin and doxorubicin and showed that if you look at the red curve, you now see that you need less of rheniaromycin and doxorubicin to get a lethal effect on your MCF breast cancer cell population. And if we analyze the number of cells undergoing apoptosis, we can see that the combination of rheniaromycin and doxorubicin would significantly increase the number of apoptotic cells in your MCF7 treated cells. Okay? Gene expression analysis also showed that combination of rheniaromycin and doxorubicin activated a different set of genes in comparison to just your single treatment of doxorubicin or rheniaromycin M. So with the interest in getting more rheniaromycin M, we actually resorted to the mariculture of the source sponge, and this was done under the DDHP project. So the sponge biomass, they source from wild population of the blue sponge. So we're sponge-titos and sponge-titas, not plantitos and plantitas. Okay? And they are transplanted And after 6 months, you can see that you have a significant change in the biomass of your sponges. So they're growing, they're happy, but it also doesn't come free because we still have um, to maintain the mariculture setup and monitor them. So we have our RA here, Lee, who's doing the monitoring. You have to clean them. You don't want them to be overrun by biofouling organisms because eventually that biofouling will affect the growth of your sponges. Okay? And at the same time, if you're doing your sponge mariculture, it will not be all about just growing the sponge, right? Unlike our plants, if we see them bloom, if we see them growing, then we're fine. But for your sponge mariculture, you have a particular target, and that would be the production of your Rhaenyaromycins. And so, with that, aside from monitoring the sponge biology and development, we also have to monitor the high-value properties of your sponge extracts coming from your mariculture. If Rhaenyaromycin M is not produced, if you don't observe anti-proliferative activity, what would be the value of your mariculture sponge? That would be probably for that sponge, right? So we have to consider how to grow the sponge, where to grow the sponge, even when to harvest, and how often can we harvest. At the same time, consider the high-value properties together with the survival of the sponge and the growth characteristics of the sponge. So we did initial trial for a year in Puerto Galera, and we started with Puerto Galera principally because the blue sponge is a lot more abundant in Puerto Galera. We also see some of the blue sponge in Puerto Galera, Mabini, but they're very tiny. They're very tiny. Okay, so we translocated some of the sponge from Puerto Galera to Bolinao, and you can see here the sponge seem to be happy. If you look at the curve A, this would be the sponge survival. Yes, both of them survived in Bolinao and Puerto Galera. B would be the specific growth rate. And yes, the sponge in Bolinao is also growing. But what would be the difference between the extracts of your sponges in Bolinao and Puerto Galera would be the high value character. Graph C would be the Rhaenyaromycin M content, as you see in the y-axis, and be the cell viability. So if you have more production of Rhaenyaromycin M, then the cell viability of your cancer cell line in graph D should be low. Okay, so it would be inverse. High Rhaenyaromycin, low HCT116 cell viability, and that's what we see here. Basically, the translocated sponges in Bolinao didn't produce Rhaenyaromycin M, but the Puerto Galera sponges did produce Rhaenyaromycin M, and you see the cell viability of your cancer cells being low when treated with extracts from Puerto Galera, but the cell lines from treated with Bolinao, they're very happy and proliferating. Okay, so that's also another reason why we decided to focus our mariculture effort in Puerto Galera. How do we grow the sponge? So what we saw in the video is the swim method. That's actually an improvement from the tube method that you see here above. That's the tube method, and the figure below, the bag method, is the one at the bottom. Okay, obviously the bag method would have a disadvantage because of biofouling. So it's a lot more difficult to maintain, and it will cost a lot more in terms of maintenance, while the tube method here, your principal consideration there would be wave action. You don't want to put it in an area where you have a lot of wave action because it can easily drift off. Okay, but in terms of survival and growth, you don't see a change in or a difference between the tube and the bag method. And the same thing with the Rayneromycin M content and your HCT116 cell viability. So the Rayneromycin, your high value characteristics together with the survival and the growth of your sponge, it's independent of how you culture your sponge. Next, how many times can we harvest? Is it just once a year, twice a year, or thrice a year? Of course, you want to harvest multiple times, diba? Just like your fruiting trees, you want to harvest multiple times so you can have a lot of output. The same thing with your sponge titos and sponge titas. We want to maximize the harvesting. So to see here would be that the survival really doesn't change much with the harvesting regime. And apparently, your growth rate, as you see in graph B, it's also higher when you have more harvesting in a year. And at the same time, Rayneromycin M content and the HCT cell viability, they're not affected by your harvesting regime, and you can actually do 3 times harvesting in a year, and that would be a lot of sponge biomass that you can now work on for the purification of your Rayneromycins. Okay, so the next question: when can you harvest the sponge biomass? Do we have the same quality of sponge biomass throughout the year? And the short answer is no. Okay. And look here, the graph. So the right that monthly average temperature and the net graph would be in A when you're circulating. B would be your specific growth rate. There doesn't seem to be any difference in your sponge survival different times of the year. Specific growth rate is the sponge seem to be not very healthy in growing in the months of May and also in January. And this coincides the time where there is a high change in temperature. So you can see that it's becoming warmer with an increase in your sea level. Now, since we had a hypothesis that your blue sponge, the quality of the extracts is affected by your temperature, we analyzed in more detail 3 sites where we saw your blue sponge University of the corresponding sponge, and this was work done by my student Jenny Manzano, spread for MSc here at MSI. And what we saw here is for all there is a very different chemical diversity between the sponges, extracts of sponges shown below, which is shown in Ceylon, compared to the extracts from biomass that's collected in stone. On all three sites, there's no change or difference between the chemical diversity or the molecules present in the extracts of this blue sponge. Through time, you see differences in the characteristics of your extracts. So let me divide over this. We look at the assembly resource of your second compounds that can break potential as anti-cancer agents. But of course, is it all that? There's different proteins also have some other potential that probably not can be fine. And so we started in the lab to develop your information assay, and this would require your well, 264 coincidences, these are macrophage cells, and in this information, we're adding LPS, or lipopolyester current. That you now measure the production of nitric oxide. So that's the primary assay to measure the change in nitric oxide or not. And then afterwards, we have to look at your screen for your sensitivity because we have to be sure that the change in production of nitric oxide would actually be because of the effect of the inflammatory pathway and not just inflammatory effect, but in fact it's not. And so we screen about 230 extracts from our chemical library for anti-inflammatory components which were done by Julie Susana. She had her thesis here on site. And so what we want here, we want extracts that would have low percent nitric oxide reduction when you treat your macrophage cells with this extract, increase nitric oxide reduction at this time. They should not have any cytotoxins. So the cell level should be close to 100. And the first extract that we pursue as the yellow sponge Neotertia. Neotertia, you can see here the oxidation, that's the corresponding yellow. It costs about 23% in NO production relative to the silver, while the solubility remained at 100%. So it's not uh, in effect due to the toxicity that the hypothesis found that the compounds produced by milk precursor should be missing production and toxicity of the compounds coming from Neopetrosia. And here you have the IC50 value. So what do you want in terms of the IC50 for NO production? The IC50 should be lower. So a molecule is a potent anti-inflammatory agent if the IC50 corresponding to NO production would be low. Okay? And for cytotoxicity, we don't want them to be cytotoxic, so the IC50 should be high. So that should be inverse. And here, the mother of all these molecules is cestoquinone, as you can see in the first bar. And we have the disubstituted adosaquinones together with the monosubstituted analogs 4 to 6. So the cestoquinones and the disubstituted analogs, they're the star molecules for a long time because they're really very potent cytotoxic agents. So the monosubstituted compounds, they're not too well recognized because they don't, it's not really a very potent activity that they show. But here we saw that they have single digit IC50 for anti-inflammatory activity. At the same time, look at the IC50 for cytotoxicity. They're not cytotoxic at all, right? So 4, 5, and 6, you can see that the IC50 for cytotoxicity is more than 10 micromolar. In comparison with cestoquinone, that's about 5 micromolar or so. Okay, so if we compare now the selectivity of your cestoquinone analogs, so you can see that maybe if we look at the selectivity index, there's almost no difference between the IC50 corresponding to the cytotoxicity versus the ones corresponding to the NO production. But what you want is something like the ones observed for compounds 4, 5, and 6. They're fivefold higher compared to the IC50 that we observe for your NO production. Okay, so we use here your compounds 5 and 6 as our model to further probe the activity of your molecules. So we didn't stop an NO production. What are the pathways that would probably be affected by your monosubstituted analogs from this sponge? What are the pathways that's affected? And we can see here, this is a gene expression analysis, and the y-axis would be the relative expression of your genes. So the genes are NOS2, which would be responsible for the production of your nitric oxide. IL-1 beta is interleukin. It's also pro-inflammatory. And NQO1, this is a pro-antioxidant. So it's an antioxidant gene. So in effect, what you want would be low NOS2, low IL-1 beta, and high NQO1 gene expression. Okay? And we can see here, if we have LPS alone with DMSO, the relative expression of NOS2 and IL-1 beta, we're at the 1,000 range. But if we decrease, if we add your monosubstituted cestoquinone analogs, we see a significant reduction in the expression of NOS2 and IL-1 beta. At the same time, we saw an increase in NQO1. So with the gene expression changes that we observe for NQO1, together with the structures of these compounds, we hypothesize that these compounds would target your NRF2 pathway. Okay? And so we look at the expression of the NRF2 gene using a specialized cell line, and we look at the activation of your NRF2. So for cestoquinone in the disubstituted analogs, you see very small change in NRF2 expression. But if you look at compounds 4 and 5, you see that you have a significant increase in your NRF2 expression. So what I would like to point out here would be that by interfacing a new assay with the chemical diversity that we have already in our chemical library, we can find new applications for otherwise old molecules. Okay, so we saw that your monosubstituted cestoquinone analogs, they would bind to the cysteine residue of KEAP1. This would lead to NRF2 translocation in the nucleus, which would change the gene expression of your IL-1 beta and your NOS2, which are pro-inflammatory, and would increase the transcript level of NQO1, and eventually this would lead to a phenotype that would decrease NO and correspondingly would indicate a decrease in your inflammation. Okay? So I shift gears a bit, and I would like to share with you my favorite organism, bias aside. But it's really more fun because it's cyanobacteria, right? So, di ba? Cyanobacteria. And they're ubiquitous organism. They're very resilient. And they're very productive producers of secondary metabolites, also known as your blue-green algae. They're present in all forms, unicellular, filamentous, and you can see they also form big blooms, and they are very successful organisms that have survived for billions of years. They don't have any defensive structures, they can't fight with you, but they have rich chemical arsenal that they can use to defend themselves. So you don't need a brain to survive, you need a good chemical adaptation, okay? So, coming back as a Balik PhD, I brought my passion for cyanobacteria here and started screening extracts of cyanobacteria, and we didn't have to go far because people all over the UP system, they already had cultures of cyanobacteria, so we were happy. And we started looking at the chemistry of these molecules and also looking at the chemical diversity and the bioassay. So you can see here, again, your HCT116 cells, you want those cells not to survive. We also did an anti-inflammatory assay using the same method as what we did for the cestoquinone compounds, and also anti-malarial activity against Plasmodium falciparum. So green would indicate high survival, red would be low survival. So obviously, no active extract against Plasmodium, so they're not anti-malarial. If you look at this HCT116 graph, you see most of the columns are red, indicating that the survival of your cancer cells would be very low with the treatment of these cyanobacteria extracts, indicating that most of them are cytotoxic. And so that complicated the anti-inflammatory assay because we see some decrease in the NO production. But that's only because of your cytotoxic effect. So we pursued the cytotoxicity, the cytotoxic extract purification. We purified several of these alkaloids, but this is a gift that keeps on giving because aside from the cytotoxic alkaloids, we also saw different peptide-based molecules that are bioactive, and the discussion of the structure elucidation would be reported by Jeremiah in a while. So, welrecyclamide, this is a cyanobactin, and aside from the welrecyclamide, which we saw in the biosynthetic gene cluster of your of one cyanobacterium, Hapalosyphon welwichi, this cyanobacteria was also a prolific producer of non-ribosomal peptides. Okay? But we did not purify in the crude extract your non-ribosomal peptides. Maybe, well, the genome would tell you what's supposed to happen, but obviously, more often than not, it doesn't, you don't see the production of the metabolites for one reason or another, the lack of your raw material, okay, the primary metabolites or certain cofactors. But certainly, the biosynthetic gene cluster of this cyanobacteria indicate the production of a non-ribosomal peptide. And that's where another student came in. Janelle Casanova did the synthesis of non-ribosomal peptides predicted from the BGC of this cyanobacteria. And this the predicted structure of this non-ribosomal peptide was closely related to coibamide, which is a marine produced by a marine cyanobacteria, and also a lactococcus bacteria. So Janelle did the synthesis of these compounds, and so from one BGC, by changing the post-translational modification from certain in certain amino acids, we got many compounds. So just by changing the stereochemistry of the tyrosine and also changing proline to hydroxyproline or methylproline, we had a total of 6 compounds. While the synthesis was straightforward, the bioactivity with the antiproliferative assay and antimicrobial didn't give any activity. So, and this highlights that, again, we're limited with what we can screen, okay? And so right now, we don't have an application for this, but in the future, when more bioactivity platforms are available, then we could, again, throw it in the assay platform. And that's where what we want to do for our marine natural products legacy collection. So in all years of doing extraction, purification, we ensure that nothing goes to waste and obtain not just the microbial biobank, but the chemical library as well. So this is a living collection. We add as we go along the way. We now have about 6,000 microorganisms, 5,000 extracts, and 237 pure compounds. So if you're a grad student willing to work, there's a lot of work waiting for you. We just need graduate students together with new assay platform. We can try to find new applications that we can do for our microbial biobank and also our extracts. And I would like to think that this would be our contribution to MSI, as MSI goes to 50 and beyond. And we will maintain this for other researchers to also use in the future. And so what I want to leave you now would be that innovations from marine organisms from the Philippines, it all starts with the biodiversity. And we're very fortunate to be in the Coral Triangle where biodiversity is really in our backyard. And so we have to be part also of the conservation effort and also as chemists, microbiologists in the field, even though we're hydrophobic, we also have to do our share in the conservation effort. Again, when we go to the field, we're taking snapshots of the biodiversity that's present. We're taking snapshots of the chemical diversity that's present. If we go a year from now, 5 years from now, 10 years from now, we don't know if we'll get the same set of microorganisms or the same set of chemicals from these marine organisms. So we're putting it in the lab, we're preserving it, and we're hoping that we'll find more collaborators to work with. Okay? At the same time, I don't think we ever went beyond the sunlit region of the ocean. So there's certainly a lot more niches that we can explore. And again, if you have more unique ecosystems, the chances of finding new molecules is also very high. So that's something that we can work on here at MSI. In terms of taxonomic and genomic analysis, I think we have a mass already of within the university and all other researchers in the Philippines. There's also already a lot of sequences already from Philippine microorganisms. We can certainly move forward with large-scale genome analysis and try to find patterns. At the same time, integrating now chemical data, looking at your genome and linking them with your metabolome. For the chemical analysis, we have done a year worth of monitoring the chemical diversity in the blue sponge, but I think we have to look at a longer term perspective. What would happen now with the chemical diversity over a period of time? What can happen to the chemical diversity with the changing conditions of the ocean? I don't have an answer for you, and I hope a graduate student would be there to answer those questions. And at the same time, doing more integration of the physical, chemical, and biological data. It's been really a fun project with the DDHP Blue Sponge work because we had to work a lot with a group of Perry and Van, the biologists, and also the interface it with the nutrient analysis. And we hope to do more of that to understand the chemistry and the biology of the organisms together. And of course, with the activity screening, we can develop more platforms. It certainly doesn't have to stop with biomedical aspects. It can be other applications. And with the current hit compounds that we have, we already did the mechanism of action studies, how they can work, how they can exert their efforts. The next step now would be going into formulation and in vivo stability. And with this, we also ensure that we protect our intellectual property by doing your invention disclosure, patent application, and with that, you can now have more industry engagement. And so with the 10 years at MSI, I'm very fortunate to have worked with such an excellent group of researchers and also received funding from several funding institutions, the DDHP Marine Component Project, the PMS ICBG, FICO Pro, and also the Picari IHIT 63 colleagues and collaborators from the Institute of Chemistry, within the MSI, biology, and all over the world. And as we celebrate the 20 years of the lab and the 50 years of MSI, I'd like to pay homage to the beginning of marine natural products and all my personal heroes in one picture. You can see here National Scientist Lourdes Cruz, Dr. Giselle Concepcion, Dr. Coke Montaño, Ma'am Rod, Sir Perry is there, Ma'am Lau is also in the picture there, and also Sir here. So certainly, they paved the way for marine natural products to thrive here at MSI and in the country, and we're very grateful for their continuing guidance to researchers like me and also my students. And of course, looking back, now looking forward, it's been a wonderful time and the most fulfilling part of the job, mentoring students at MSI, at IC, and it's been fun working with such a lot of brilliant, hardworking students from 2014 onwards. So thank you very much. I'd be happy to answer your questions. Lang.
So I think it was last month. I attended this conference at Iloilo, representing Lil, and it's really amazing that we see a lot of major advances in this field, but I think it's kind of well, I think the idea is that although we still are leaders in this field, sort of we need to make sure that we maintain that leadership somehow. And I'm really glad to see all the developments that have been accomplished in Lil's lab. So if there are any questions, I think Lil is ready to talk the floor now.
So, curious ako pagdating na sa, well, we're now talking about like culturing the blue sponge and so on. So since these are, like you said, secondary metabolites, so you saw the difference between the Bolinao and the Puerto. So how would you ensure that you can get those secondary metabolites and like, and grow the source material and still be able to get the same secondary metabolites?
Yes. So thanks, Ma'am Lau, for the question. So that's why for the initial setup that we had to monitor the conditions as well as the production. So more or less, and we also chose the site where we see a large population, natural population of the sponge. So they would be happy there, growing there. But surprisingly also, in our subsequent mariculture setup, we tried to simplify things, but apparently there were differences in the production of Rhaenyaromycin. So even if it's in the same location, but some tweaks to simplify the mariculture method and try to avoid predation, the sponge doesn't seem very happy. So it's also not all about just the sponge itself growing, but also trying to maintain the relationship. Just like boys and girls, they also need relationships to be happy.
So synthetic the way to go eventually?
The synthesis, even for the echinocydins, no, synthesis is not an option because it's too complicated. So what they're actually doing would be to produce an analog from a microbial source and then modify that for via synthetic route, a one-step synthetic route. Because doing the total synthesis from scratch and scaling that up to gram level, it's not very feasible and it's difficult.
I'm really fascinated how you started to talk like an ecologist and very convincing.
It's from the association, Helen.
So, well, just a very short human interest aspect of this. So I was struggling with Miguel Ascuna, trying to understand that chemical structure from the blue sponge, because Giselle was adamant that's what he has to study, right? And then and then Van told me, naku Helen, ang hirap talaga i-culture.
Yeah.
Okay. So I was really so happy to see the video of Lee, whom I
Yeah,
also
yeah.
know. He has for plants it's a green thumb, but for Lee, what do you call it? He can grow sponges.
A blue thumb.
Yeah,
It's a blue blood.
yeah. So it's amazing. He got to get them to grow that big. All right, but as an ecologist, I suspect one of the secrets is the site.
Yeah.
Yeah. Do you think there is a chance that the sponges, as they're growing, they're exchanging microbes with the surrounding water? Could the site be very unique because it promotes sponge abundance? And could the microbes be sort of flowing in the surrounding water, and they might exchange, they might enter sponge cells,
Yeah.
and establish themselves to produce the secondary metabolites. And my accompanying question and concern is the site has to be preserved.
Mhm.
So, so if you want the sponge population to continue to thrive, we have to prevent pollution,
Right.
keep the water quality.
Yeah.
Okay, thank you.
That's why when there was the oil spill, we had the site also checked by staff from Cradle and luckily didn't get affected by the oil spill. Regarding the question whether there could be an exchange of microorganisms within the sponge, well, for other sponge species producing Reiniermycin, they showed that there's an obligate symbiont. It's an obligate symbiont. But certainly, if, for example, for the transient microbes, could be exchange, and maybe this would be where Dr. Roy's group would come in because they also did microbial population studies on the blue sponge. So, advertisement, the talk of Dr. Roy is May 5.
Okay. Raf, please alert me if there are questions from Zoom because I can't see anything from here.
Yeah.
Wala pa, okay. Other questions?
Okay, done.
There was a post I read about heat tolerant corals breed heat tolerant corals. Would the same thing apply to sponges in terms of highly productive sponges breed highly productive sponges?
We actually don't know. Don't have an idea with as far for our work we use the same mothers or source from the natural population in Puerto Galera, so you can think of it that way. If it's productive, because the microbial endosymbionts are already there, so the biosynthetic gene cluster should be established there. So then you would say that that could determine the productivity of your blue sponge in terms of their natural products production.
I have a follow-up question. So, uh, wait, no. So in our total ecology class, basically the production of secondary metabolite is primarily driven by competition for space. So we know that sponges don't want any sponges or other species impinging upon their personal space. So doesn't mariculture uh, wouldn't mariculture affect that because it's you're basically providing a solid condominium for sponges.
Okay.
Yeah.
I'm happy Eden listened in my class. Um, but so for the blue sponge, we also did a survey in terms of whether there's an association between the blue sponge and the corresponding benthic ecosystem. But actually, there's no correlation. So it doesn't matter where they are, if it's a rubble, a dead coral, or so on. So in this case, putting them in an artificial setup, they should still be happy. But if you remember another story in class that we discussed, the kahilalide, aside from competition, there's also predation. And in terms of kahilalide production, it seems that a predator is necessary. And that's my personal hypothesis for this blue sponge also. Yeah. So, for example, for Bolinao, there doesn't seem to be a natural partner for your blue sponge, but they're certainly abundant in Portugalera. So there's a partner. It's not just only a partner between the sponge and the microorganism, but they also see also a macroorganism that has a relationship with the sponge. So it's a tripartite relationship.
Okay, so maybe just follow up to Eden's question. Um, since the sponges are actually growing in this whatever contraption that is, it would appear like it's not competition for space or other sponges. So, but would you think that the production of reneromycin or related compounds are responses to stress, and what could constitute stress in that kind of environment?
I think it's complicated relationship. Well, my hypothesis would be that it's not really predation per se, but more of like also a symbiotic relationship between the sponge and the macroorganism. The macroorganism may be hijacking some of the sponge compounds so that the sponge now has a natural role or natural reason to produce the molecules for the partner macroorganism. And consequently, the sponge is reliant on an obligate symbiont as the biofactory to produce all this. Helen.
Organism is a picture.
The microorganism nudibranch.
It's a nudibranch.
It's a
Are you
new.
keeping that as a trade secret or?
No.
So you observe this nudibranch in that site and not in others?
And it's abundant in Portugal.
Oh,
And that's a limitation for our study because we never did a survey for the nudibranch, right? We did fish, but just personal observation, but not like a continuous observation. But if you follow the the story of Kahilite, Alicia, and its partner organism, it's almost the same story.
conserve the population of the nudibranch as well.
Yeah. So because if the microorganism is an obligate symbiont, so if you transfer it to Bolinao, then it should persist, and they should be happy to produce the rhaenyramycin, but apparently not. So there's something missing in the equation. Well,
Okay for you to divulge what type of nudibranch it is. No, because based on our researches, if you recall, you mentioned about the Elisha and its coloration of having its chloroplast being absorbed, right? So would the color matter, or probably whatever relationship it has, we can identify it in the future?
yeah. So it's if you look at pictures of the blue sponge, it's always associated with the Juruna fernubris.
Thank you.
And they're happy munching on our blue sponge. Thank you very much. Thank you, everyone. And now I begin with our group's sharing of our um, accomplishments. So we have lab alumni and also current members of the Marine Pharmacognosy Group to share their research on natural products. And so for our first speaker, we have Mr. Noel Lacerna. He's a graduate of MS Chemistry in 2017 and is currently a PhD candidate at the University of Utah. And he will talk about developing autocatalytic macrocyclases for peptide library generation. So, Noel, floor is yours.
Okay. Hi, everyone. Can everyone hear me?
Noel, maybe you can go closer to the microphone. You're
Okay.
just a
Hello.
little bit choppy.
Hello.
Be better. Sige, go ahead.
Okay. So, hi, everyone. My name is Noel. Long time ago, I'm also a research assistant in Marine Science Institute for about, if I remember, about 7 years. So now I'm a PhD student here in University of Utah. So our lab is interested on biosynthetic pathways on how small molecules are made. So today I'll be discussing about how to develop enzymes, particularly macrocyclases for cyclic peptide library generation. So cyclic peptides are abundant in nature in that they have and that they have been used as a drug for the last decades. So example of cyclic peptides are vancomycin, cyclosporin, romidepsin, griselimycin. So as the name suggests, these compounds have macrocyclic structure. So macrocyclization provides conformational rigidity, and this provides pharmacological properties such as protease resistance, specific affinity to receptors, and even membrane permeability. So recently, macrocyclic peptides have been increasingly identified to be a new class of drugs because they have this property combination of biologics and small molecules. So biologics, because they are known to be highly selective, and similar things can be mimicked with these cyclic peptides. So small molecules, on the other hand, have these properties such as being cell permeable and stable, and that some peptides, cyclic peptides, have also these activity. So with these properties and other considerations, cyclic peptides seems to be an interesting drug modality. And moreover, development of methods for peptide cyclization seems to be worthy. So peptide cyclization has been a common interest for the longest time, and numerous approaches have been published. So typically, it's most of the time it's purely synthetic or semi-synthetic. But our lab, which is interested on genetic approach, we are more interested with a genetic approach towards peptide cyclization. Why? This is because we can create combinatorial libraries through genetic randomization, which can be coupled with high throughput display or like screening platforms. So among this is mRNA display, which can be coupled with in vitro translational methods. So these approaches have been very productive and have been useful on identifying peptides as potent inhibitors on disease relevant proteins. So, however, the most coveted methodology that we want is producing cyclic peptides that can be randomized inside the cell. So, using this approach is very helpful, as cyclic peptides can be tested in a cellular context, and at the same time, it can be coupled with a selection system or like a phenotypic screen. So right now there are only a few methods that were discovered, and one of these is called Cyclops, which stands for split enzyme mediated circular ligation of peptides and proteins. So enzymes are self-excising protein domains that process to link their flanking sequences by a peptide bond. So construct are two enzyme domains, N and C terminus, and then you have a amino acid sequence that you want to cyclize. So these would undergo series of rearrangement, and what you have is a cyclic product and excised enzyme domains. So the problem with this is that these approach sometimes most of the time leads to causes like incomplete cyclization, and what you have is a mixture of cyclic and linear product. So my lab in here is that we are interested on developing a more efficient method that could circumvent these problems. So our lab has a long time interest with a particular biosynthetic pathway called RIPS, which stands for ribosomally synthesized and post-translationally modified peptides. So the word RIPS, so that means that these pathway functions as a typical proteins, like a typical central dogma of proteins being made in the ribosomes. So a common architecture for these pathways that you have your post-translationally PTM enzymes, the modifying enzymes, and a small structural gene called precursor peptide. So if you look at these precursor peptide, this is just very small, probably around 30 to 100 amino acid. It contains multiple recognition sequence for each of the modifying enzymes to bind, and a core sequence which is underlined in red. which is then being processed by these modifying enzymes. And then after multiple enzymatic reaction, what you have is a mature compound. So in the precursor peptide, these recognition site, these green, orange, and blue, are highly conserved because they act as a clue for the enzyme recognition. So one classic example of a RIP enzyme is called ALIND, and a common feature for these type of enzyme is you have two domains. A domain for substrate recognition, which recognize these recognition sequence from the precursor peptide, and if this positions the core in the next domain, which is specific for only for modification. So note that there is no key interactions with this. So this feature makes it a lot of RIP enzyme to be highly site specific and highly promiscuous. When I say highly promiscuous, it means that it can process a lot of different sequences. So one RIP pathway that we are interested are called cyanobactins. So cyanobactins are these peptidyl natural products from cyanobacteria. They possess a lot of biological activity such as anti-cancer, anti-HIV, methylation, whatsoever. So some common features from cyanobactins is that they are often cyclized, and then sometimes these heterocycles are installed in the backbone, and that sometimes they are often also decorated with isoprene units. So this pathway is what we call as a diversity generating pathway, because they are capable of making multiple products compared to some metabolic pathway which specifically work and produces one product only. So how are these different compounds generated by a simple by the same pathway? So here are three precursor peptides from the same pathway, and you can see that they are basically the same except for the core sequences. So the enzymes that are responsible for this pathway are highly promiscuous, enough to process all of the core sequence and generate variety of products. So besides from these pathway, there are also a lot of other pathways capable of doing or generating a lot of products. So these pathway can produce millions of cyclic derivatives. Now, because we are more interested in how on peptide cyclization, the enzyme necessary for the cyclization step is what we call as G enzymes. So they function as macrocyclases. So they are typically your serine proteases, meaning they have this catalytic triad of serine, histidine, aspartic acid, and you have another feature called capping helices. So the first process for this G enzyme is that your first core sequence is that it needs a turn-inducing element, which is often a heterocycle, such as a proline or a thiazoline ring. So in this way, it would allow the N-terminus of the core sequence to be near that acyl enzyme intermediate. So the first step is that you have your core sequences, and then it would position, and then the capping helices would position the core sequence inside near to the active site, and then the acyl enzyme intermediate would be produced, and then the N-terminus gets deprotonated, and what you have is peptide transpeptidation, and what you have now is a cyclized peptide. So G enzymes are considered to be high fidelity, meaning they can only produce cyclic peptides, and that they are broad substrate tolerant, meaning they can produce a lot of cyclic peptides with different sequences. However, the problem is that these enzymes are considered to be very slow, with turnover number about approximately 1, which is basically slow. Another problem with using these enzymes is that before cyclization has to happen, it requires a minimum of 2 steps before the core sequence getting cyclized in the native pathway. So general problems in a lot of enzyme reactions, or specifically with G enzyme, is that with this you have slow enzymatic activity. You also require higher concentration of substrate, and then you have some issues with solubility of the substrate and dimerization issues. And in vivo, if you want to use it, the substrates are prone to proteolytic degradation because you only have about 50 amino acids. And then at the same time, if you want to do it for in vivo, the enzymes and substrate should be in the same space. And at the same time, the native pathway produces a lot of intermediate before cyclization has to happen. So the question is, how can we engineer these enzymes and utilize them for both in vitro and in vivo applications? So we think that the best way to do that is to design these enzymes by fusing the substrate with the enzyme. In that way, you are limiting the movement of the substrate and making it in a first order. And what you have is an enzyme that's autocatalytically cyclize the core sequence, and what you have are cyclic peptides. So the cyanobactine pathway, there's a lot of cyanobactine pathway, and that they produce a lot of natural products with different combinations. In here, what we first use is this called Pag-G enzyme from perennilageramide pathway. So in this case, the requirement for the core sequence to be cyclized is that it must have a proline at the C terminus of the recognition sequence. So we can just proceed with putting a linker and hoping it would work. So we wanted to design an enzyme that can be purified and then optimized for the activity. One of the G enzyme has already a crystal structure, so we try to approximate the distance between the N terminus of the and then the catalytic site. So we measured that distance and then use it to generate how long the linker should be. So in this case, we use 2 common linkers in fusion proteins. You have this 4Gs and 1S with a different iteration in a GSG linker. We design about 8 different types of proteins in here. So now these 8 proteins, it might sound confusing, but it's basically just a single protein with each specific with some specification. So take note that in this form, Pag-G enzyme can only cyclize the core sequence, about 6 to 8 amino acids, and therefore if you add additional sequence on the N terminus, it would not cyclize. So if we add an N terminal protease, what happens is that you would cleave this you would cleave these leader sequence and then a recognition sequence for these for these N-terminal protease. And what you have is only this protein, the core. In this case, what happens is you'd free the N-terminus of the core sequence, and this would automatically cleave and then cyclize. And so what you have is a so what you have is a cyclized product which you can be detected to mass spec. So we were able to obtain purified his tag proteins as shown in this denaturing gel, and then run the assay. So we are measuring the cyclized cyclic peptide formed in a mass spec, and from this figure we can observe that this combination, the linker GSD linker with these with this size produces the amount of cyclic peptide, highest amount of cyclic peptide. So now using the data from the in vitro study, we then perform a heterologous expression. We just wanted like the E. coli to produce it without any interference. So we removed those additional sequences, and then just design these enzyme, and then we call that autopag-G. So you have these core with this QAY LGIP LP. And then the recognition sequence, which is this FAG-DDAG, and then the linker. And then after that, we transform it in a BL21 E. coli, cultured it, and then after that, check whether the compounds are being produced. So now from MS data, we can see this peak from the acetone extract of our E. coli culture, indicating that our construct is working in vitro. However, upon further examination of the extract, we can also observe cyclic peptides with methionine and an oxidized methionine. So where does this methionine come from? So remember that in protein translation, we usually start with the start codon, and that start codon, the ETG, is translated into methionine. So these suggest that what we have now is like a competing reaction between typically these methionine is cleaved after the protein is made, but in this case, once the protein is made, the methionine is being included in the cyclization. So what you have is a competing reaction between methionine excision and cyclization, which means that we have solved one of the problem with these enzymes. We have made the enzyme to be to have faster. This means that the enzyme has now faster kinetics. However, the problem is that while we made the enzyme faster, we are now producing three different types of cyclic peptides, which is not good if you want to design a high fidelity library. So what we want is still a faster enzyme, but with high fidelity. So in this case, what we want is to solve this problem by slowing down the enzyme, somehow slowly making the enzyme a little bit slower, or by using an additional control towards cyclization. So in this case, we use another G enzyme from cyanobactin pathway called PAT-G enzyme. In this case, you require a thiazoline ring for cyclization. So typically, the core sequence for this is that you have a cysteine on the recognition sequence, on the C-terminus of the recognition sequence, and then this is being converted into heterocycle into a thiazoline ring. So to do that, we utilize another PPM enzyme called RS1-3D, and this converts the cysteine, in case this one converts the cysteine into a thiazoline ring using ATP. So recently, a group showed that these enzyme modify can modify cysteine in thiazoline ring in the C terminus on the loops and flexible regions of end cherry, provided that there is enough space for the enzyme to interact, and that the recognition sequence is embedded on the right before the cysteine residue. So in our case, we repurpose this enzyme to be some sort of protein activator in a protein in context. So the thiazoline ring is in the core sequence is just is the requirement for before peptide cyclization. So using the optimized conditions that we use in the initial step, we design another protein construct. We call that as auto-PATG, using the PATG enzyme, and you have these core sequence. So you have a soluble protein. So what happens is once you get the protein, you reacted with this another enzyme called RS1-2D, and then the cysteine gets converted into thiazoline ring, and it would automatically cyclize, producing the cyclic peptides, and then the cleaved G enzyme. So after 24 hours, we observe that peak, the cyclic one, and we also observe a dimerized form of these, but this one is only on these core sequence. So we tried other different core sequence, such as a mutant. So we replaced the leucine into valine, and then we also design an 8 amino acid core, and we can see that peak at 8.6 minutes, which shows the cyclized product. So in this case, we successfully make protein construct that can cyclize core sequence on its own. And so we think that we think of applications on these technology, and one thing that we could think of is, what if we can sorry. So we did that also. We did that sequence also by heterologously expression in E. coli using those two enzymes. You have these autophagy, and then you have an RS1-2D. So these would be expressed for 5 days at these conditions, and then after that you get the pellet, and then you extract it. And from here you can see this compound. So we think of an application of this technology, and we think that maybe why not build a cyclic peptide library. So we develop an ectopeptide library through mutagenesis. So in this case, using that PRSF vector, duet vector that expresses both two enzymes, we randomize every core sequence from P1 to P7 position, using 9 different amino acids, and that would theoretically produce about 9 raised to 7, or about 4.8 times 10 to the 6 potential cyclic peptides. So because we cannot sample everything, so we just get 100 of them, sequence them, and then express them, and then after that we were able to check them in mass spec whether they are being produced. Now, out of the 100 mutants, we just get only 94 of them, but out of that 94, we get about 51, or about 54%. And if this numbers holds up to this library, we are expecting to get about 2.6 million distinct cyclic peptides. And just an example, these are some of the sequences that were cyclized. So we check those sequences and see if there is a particular trend, and we noticed that all amino acids that we put in this library is that are all accepted, but not equally. So as you can see, the enzyme has a particular preference to small amino acid, and that aromatic amino acids are less accepted, particularly at the ends of the core sequences. So you can see that in non-cyclic sequence, you have the phenylalanine and then the tryptophan are not accepted fully. But in these experiment or in these observation, we noticed that all amino acids are accepted, but not equally. So because we noticed that that a lot of these cyclic peptides are being produced in a lot in the mass spec, we tried to isolate one of these mutants. We use this sequence TAYWPWIC as an example because you can purify them well, and then you can see them in HPLC. So we just we use the plasmid containing these core, cultured about 12 liters of E. coli, and then followed by centrifugation and then extraction of the pellet. And then after that, purified it with series of chromatography and reverse phase HPLC. And what you have is this peak, which is your compound. So to validate whether this is real, we do NMR, and then the proton NMR looks like this, and then after that, we try to fully confirm it using 2D NMR, and what you have is confirmation that what we have is these cyclic peptide. So we use this as a additional as a standard, and we use this to determine how much of the cyclic peptide is being produced. From here, using the standard addition curve, we can get about 17 milligrams per liter of cyclic peptide from a liter of culture, pre-purification. And then when we try to position this in the library, we notice that the amount of the cyclic peptides being produced are basically random, and that it tells you that the production is heavily sequence dependent. But you can see that there are lots of cyclic peptides being produced, more than what we have isolated. So this tells us basically that method is scalable, and that this approach can produce sufficient amount of cyclic peptides for pharmacology. So in summary, we have developed a simple, efficient, and scalable methods that can produce cyclic peptides, cyanobactin cyclic peptides, both in vitro and in vivo, and that we use this technology or this approach to generate a peptide library. So for future directions, if we wanted to further push through this technology, we can develop our tailored library for a specific assay, either in vitro or in vivo. We can do like a practical access to cyclic peptides, or we can use this development as a drug delivery strategy. So our approach can also be tailored or can be in combination with other enzymes from other RIP pathways. So what you have are cyclic peptides with more functionalities, such as like epimerization or like N-methylation. So with that, I'd like to thank my lab here here at the University of Utah, my boss Eric Schmidt, this one, and then all the members of the lab. And yeah, so here I am. When I was a research assistant at UPMSI, it was fun times, and it feels like I was really trained before going here in the US for PhD. So yeah, that's all. I'm happy to answer your questions if you have. Thank you.
Thanks, Noel, for that presentation, and certainly very nice work from the group of Schmidt. And again, pushing forward cyanobactin synthesis. So we may have time question for maybe 2 questions. Any questions for Noel? So I guess my first question, Noel, you focus on octopeptides, right?
Yes.
But cyanobactins would also have your smaller or uh, bigger ring systems, macrocycles, right? So, but for your enzyme design, is it how much tolerance would it have in terms of your ring size?
So far, we can design about in these Ph.D. What is known is that it can cyclize about 6 to 10 amino acids, so it can probably work with that ring size. Um, I know that these enzyme has also it can also there's one study that says that it can also cyclize up to 22 amino acids. Probably it can also be done. So it's just like it's just a matter of how you want to design the library. But I think it can be done like 6 to 10, or you can extend it into a bigger ring size. But yeah, but it depends on
Okay.
how what do you want for generating the library. And at the same time,
Okay.
so I'm just using two enzymes. We just use like two enzymes. So the cyanobactin has a lot of other G enzymes. So whatever the problem with one enzyme can be another can be so uh, can be supported by a different G enzyme with different substrate preference. Yeah, hope I answered
Any
questions.
other questions? Yes. Thank you. Any other questions for Noel? No questions in Zoom? Okay. With that, thank you, Noel, for your presentation. And we certainly hope for your success in your PhD, and hopefully you can visit us here in the Philippines soon. Thank you, Noel.
Thank you.
So thank you. So we now have we divide the presentations from current members of my lab into two. First would be the biodiscovery, and we have two speakers for that, Jeremiah and and Jackie, and then we have the new developments, new methods in natural products, which would be presented by Ziljan and Sabrina. So, may I call on now Jeremiah? And Jeremiah finished his MS Marine Science program last year, and he will talk about structure and bioactivity of well-recyclamide A from an extremophilic cyanobacterium.
Good afternoon. I am Jeremiah, and I will be presenting about our progress on the structure and bioactivity of rhodocyclamide A from an extremophilic cyanobacterium. So, cyanobacteria, as mentioned by Dr. Lilibeth, are photosynthetic prokaryotes and they are one of the oldest organisms on the earth. And they are they are highly optimized to produce different biochemical weapons. Called secondary metabolites, which used to defend against different grazers. So the lecithin-10, for example, is a potent cytotoxic compounds produced from cyanobacteria that can kill cancer cells. And the synthetic analog monomethyl auristatin E was developed from this compound, which was then developed into 5 FDA approved drugs in the market for cancer therapeutics. And so in our effort to the, contribute to the natural product discovery, we prioritize a cyanobacterium that was given to us by the group of Dr. Cao from the Institute of Biology, UP Diliman. And this cyanobacterium was actually showed promising compound based on its chemical signatures. So this cyanobacterium was collected from a mine tailing from Pog River in Marinduque. And this river is highly contaminated with copper because of the Mar copper mining disaster that happened in 1993 and 1996. And so the cyanobacterium was also evaluated for its capability to bind with cadmium metal, which is very interesting in biotechnological applications. So in one of our silica fractions, I was able to get a good HPLC profile as shown in your left. And among these peaks, using HNMR techniques, I was able to, the peak eluting at 29 minutes correspond to an unknown compound. And the HNMR actually suggests that this compound is a peptide-based structure. So based from its chemical signals, like the alpha protons at 4 to 5 ppm, in tandem with the exchangeable protons at 7 ppm. Also, there are a lot of upfield chemical shift corresponding to your methyl groups. And with the use of different NMR techniques such as proton NMR and two-dimensional NMR techniques, we were able to elucidate the planar structure of the compound. So here you can see that we named this compound as well cyclamide A, and there are 6 amino acid residues here, that isoleucine, 2 valine, 1 proline, and then there are 2 thiazoline moiety. And so here are the 6 alpha protons, the 3 amide protons, and the 6 methyl functional groups. Since cyclamide A is a peptide, we also did fragmentation analysis to further confirm the planar structure of our compound. So this figure on your right is a fragmentation spectrum using our Water-Sivo G2Q-TOF high-resolution mass spectrometer, which is tested in Institute of Chemistry in UP Diliman. So the fragments are color-coded, and for example here, the fragment corresponding to color green, the 510, is actually due to the loss of your proline residue, while your fragment at 480 is due to the loss of valine. And then we also have the stable fragment here, the purple one at 383, which corresponds to the A2X ion due to the loss of both valine and proline. So this analysis corroborated our planar structure from NMR techniques. And now that we know the planar structure, we also wanted to study the absolute configuration of this compound. And because for NMR techniques, we can only determine the planar structure, while for mass spectrometer, we can only determine either the fragments, the sequence, or the chemical formulas
of different biomolecules. But in general, we need to get the absolute configuration because it is a critical determinant of the bioactivity of that biomolecule. And actually, we can use X-ray crystallography to do the absolute configuration as well as structure elucidation. But there are problems with using this type of techniques. One is that your compound should be crystallized before you can proceed with the analysis. And a lot of compounds are not actually can be crystallized to proceed with your X-ray crystallography. So an easier way to do the absolute configuration is to do the stereochemical analysis techniques using derivatizing agent. So for this one, we can use the advanced Murphy's analysis using the compound FDLA or FDAA. So here they use the FDAA and then they reacted the D or L amino acid to get these two diastereomers. So using your D and L amino acids, you cannot really separate the two amino acids using your ordinary C18 column. But by using derivatizing agents such as FDLA, you can now use your C18 column to separate the D configuration versus the L configuration of your amino acid. But although this is ideal, it is not really usable for compounds with a small amount because you need a lot of compounds to react with your FDLA. So with chirobiotic tag column, you can bypass the derivatization by using this column because it can readily separate D and L amino acid. Usually the L amino acids elute faster than the D amino acids. So to establish the absolute configuration, we hydrolyze the compound using 6-normal HCl for 18 hours to liberate the proteinogenic compounds or the proteinogenic amino acids. So here we are only looking at isoleucine and valine because these are the ones with your standards. But for these two compounds, they are conjugate of proline and thiazoline, while this one is a conjugate of valine and thiazoline. So there are no standards to analyze the configuration of these two. So with that, using our chirobiotic tag column in tandem with multiple reaction monitoring mass spectrometry, we were able to analyze the standards of isoleucine using the L and D and allo isomers of isoleucine, as well as the valine, the L-valine and the D-valine. So by running the hydrolyzed compound or the hydrolyzed cyclamide A and then comparing the retention time of these amino acids, we were able to identify that the isoleucine is in L configuration as you see on the isoleucine standard, and then the first valine here is also in L configuration. Although there is a small D-valine from our hydrolyzed standard, we hypothesize that this is due to the epimerization because of excessive heat used by the hydrolysis reaction. So in order to liberate this, the thiazoline moiety, we bubbled the compound with ozone or the ozonolysis reaction to get this type of molecule. So the thiazoline was supposedly converted to the cysteine moiety or the cysteine residues. However, since we are using high oxidizing agent or strong oxidizing agent, peroxide in formic acid, what we'll get or what we really got is the sulfone derivative, or we call it the cysteic acid. So also we hydrolyze after doing the ozonolysis, we hydrolyze the compound to get all the amino acids. After that, again, we did the chirobiotic analysis, but this time we use proline standard to get the proline configuration and then the other valine configuration. And we use a different method or gradient because L-proline and D-proline is not eluting at 10:90 methanol in 10 millimolar acetate, ammonium acetate. So the ozonized and hydrolyzed compound showed this chemical or retention times for valine, isoleucine, and proline. And so we were able to annotate the remaining the amino acids proline to be L configuration, while the second valine here is also in L configuration. However, although the chirobiotic tag is useful, it cannot really distinguish between the cysteic acids, the D and L cysteic acid. So we still have a problem for these two thiazoline moiety here. And so what we did is we go back to the advanced Murphy's analysis, and we react the LFDLA with our two standards, the D and L cysteic acids, and we got this standard here for L cysteic acid versus D cysteic acid. And then when we analyze the the hydrolyzed, ozonized and hydrolyzed well-recyclamide, we also get a mixture of D and L amino acids. So what does it mean? It tells us that there are three possible things happening here. Either you have the all L configuration, but due to excessive heat because of ozonolysis and hydrolysis, you get epimerization leading to your deconfiguration, or either the first thiazoline has the D configuration and the second thiazoline has L configuration, and the third is that the first thiazoline has L configuration and the second thiazoline has the D configuration. So in the end, we use total synthesis of well-recyclamide to show the absolute configuration of thiazoline, and this was done in tandem with our collaborator, Dr. Tauye from Peking University. So what they did first is the LL configuration. So with this, they compared this synthetic synthetic LL NMR with our natural product, and they realized that it's not overlapping. So it means that there is another configuration for the thiazoline moiety, and so they did another synthetic route, which is that D-thiazoline on the first thiazoline and L on the second thiazoline. And here the NMR overlap with our natural product, confirming that our natural product have the absolute configuration of all L except for the first thiazoline moiety or residue. And then as mentioned by as discussed by Noel Lacerna, so well-cyclamide A is actually a type of cyanobactin, and cyanobactin are metal binding compounds, and usually these are octapeptides or more, and hexapeptide like well-cyclamide are less represented. So usually they bind with two. The octapeptides bind with two metals. For this one, two copper metals. And so with the help of Kevin Davis and Sabrina Malto, we devised a titration experiment to know whether well-cyclamide A can bind to metals such as copper 2+. So to determine the chelating property of well-cyclamide A, we titrated increasing amount of copper 2 plus, and the resulting WEA copper complex was then detected using our high resolution mass spectrometer. So for the control, we only detected both the well-recyclamide M plus H and the sodiated analog. But with titration of 2:1 molar ratio of copper, so copper has a mass of 63 Dalton, while our well-recyclamide has a mass of 578. So 578 plus 63, we were able to get this peak here, which is 641. And this suggests that we were able to, that well-recyclamate A was able to chelate copper 2 plus. And then with increasing concentration of copper 2 plus, 1:1 as well as 2:1, it tends to chelate copper 2 plus in a 1:1 ratio. So we also injected the solution after 2 weeks in order to know whether the metal complex is thermodynamically stable at 1:1 ratio. And for your typical octopeptide, again, cyanobactines are usually bind in one peptide is to 2 copper 2 plus or 2 copper metals. But for our hexapeptide, we can only get the 1:1 peptide metal complex. So our results can be corroborated or correlated to the habitat of this cyanobacterium, which was found in copper-polluted water system. So we hypothesized that this cyanobacteria is being produced to protect the cyanobacterium and to thrive in Mugpog River. So despite having high amount of copper in the river. So copper, as we all know, is used as an algicide. So because it can interfere with photosynthesis and cell division. So for photosynthesis, it can replace the magnesium, which is the metal of chlorophyll to function for photosynthesis. And for cell division, high intracellular copper 2 plus can induce apoptosis and oxidative stress. And then we also tested the well-recyclamide against cancer cells, but we got at eight different concentrations, but they all got high percent cell viability, meaning it is not cytotoxic against this mammalian cell line. And so with these results, we then filed a patent in 2020 for the application of well-recyclamide A as a potential non-cytotoxic metal binding compound for different copper-contaminated systems such as mining areas. And so just an acknowledgement for the Balik Ph.D. Funding of the OVPAA, and then the National Magnetic Lab of the state of Florida, and our collaborators in University of Florida.
Okay. Hold your guns for your questions, and we'll have the questions at the end of the 4 short talks. Our next speaker is Jacqueline Peran, and Jackie will talk about from genes to phenotypes, mechanistic insights to oxolipin inhibition of biofilm formation in Staphylococcus epidermidis.
Hello. So, allow me to present to you our study entitled From Genes to Phenotypes, the Mechanistic Insights of Oxalipins Inhibiting the Biofilm Formation in Staphylococcus epidermidis. So, as mentioned by Ma'am Lil a while ago, in her previous talk, Lacerna's paper described this shipworm inspired compound characterized for having an anti-biofilm property. So from their initial SAR assessment, it showed that the epoxide moiety renders this activity. However, several questions still remain unanswered, and we call it the what, why, and how problem. So, for instance, what structural features of this epoxide moiety gives this bioactivity? What specific cellular functions does it affect? Why does oxalipin perform selective biofilm inhibition? And how does it specifically inhibit biofilm in Staphylococcus? But in order to answer this, we must first understand the biofilm life cycle. So typically, it starts with the reversible phase, wherein your free-floating planktonic cells, so your free-floating planktonic cells start to adhere to form a monolayer. So subsequently, they produces extrapolymeric substances or EPS, which now which leads to the formation of a mature biofilm. So this EPS usually consists of polysaccharides, proteins, and other compounds which modulate the biofilm environment. This encapsulation also reduces the efficacy of your antimicrobials because of their low penetration rate, which enhances the antimicrobial resistance. And the EPS regulation in Staphylococcus is facilitated by the ICA operon. So later on, you will again see this ICA operon. And going back to the cycle, as the biofilm matures, it undergoes structural rearrangements with which uh, which uh, which eventually may disperse and cause the release of planktonic cells and microcolonies, which can now colonize new environment. So another essential determinant in biofilm formation is the quorum sensing or the communication machinery of this bacteria. So unlike for us in humans, we use verbal routes in order to communicate, but for the bacteria, they uses chemical cues in order to talk to each other. And one example of this chemical cue is the diffusible signaling factors or DSF, which as you can see, it somehow resembles the terneuroic acid presented a while ago. So this DSF can actually affect various virulence factors such as exotoxins, hemolysins, the transition from planktonic to a mature phase, as well as pigmentation, which is shown here on the rightmost figure. And for the quorum sensing, the pathway involved is the AGR system, which we will touch later on. So in order to solve our what, why, and how problem, we designed this set of experiments, including the SAR, which wherein we utilizes other structural analogs. So here we used synthetic analogs. So for the temporal, we want to know where in the biofilm life cycle does our compound targets. And for the phenotype, we selected different assays which can be corroborated by gene expressions and in silico studies. So first, we want to evaluate the underlying functional motifs important in biofilm mitigation. So in this table, we present three pathogens. So two are gram-positive, the S. epidermidis and the Staphylococcus aureus, and one gram-negative, coined by Pseudomonas aeruginosa. For the analogs tested, we have different sets, which include saturated fatty acids, stearic and palmitic acids, compounds 1 and 4, which have trans-epoxide moiety, compound number 5, which has a cis isomer as compared to compound 1, and the unsaturated trans-bearing oxalipin. So based on the result, what we can see is that all of the analogs are ineffective against gram-negative Pseudomonas aeruginosa, both for biofilm and the planktonic cells. But if we are to look for the gram-positive bacteria, we can see that they have selective inhibition against biofilm of S. epidermidis and S. aureus. In terms of potency, the least or the non-effective analogs belong to saturated fatty acids, while the most potent ones belong to those having trans-epoxide moiety. This is followed by the cis isomer, which is twofold less potent than the trans isomer. While the unsaturated oxylipin is the least effective among all the oxylipins tested. So from this SAR, we can say that the configuration of the epoxide is the critical factor which renders the antibiofilm by activity as compared to the positional effect of your functional group, which is somehow not significant. On the other hand, saturated fatty acids are not effective as biofilm indicators. And lastly, we observed selective inhibition towards gram-positive bacteria. And so from this SAR, we can now prioritize compounds 1 and 4 since they are the most potent. So these compounds now will be used for the succeeding assays. So for the temporal kinetics, we want to validate the selectivity index of the oxylipin against the biofilm formation. So here, we present two line graphs. So the left one monitors the planktonic growth during the 24-hour incubation, while the right one quantifies the biofilm mass across the incubation period. So what we can notice is that the planktonic cells are still alive upon the treatment of oxylipins, as shown by the orange arrow. Unlike the chloramphenicol, which is shown by the green arrow, where there are no planktonic cells alive. So this is expected for chloramphenicol because chloramphenicol is a known antibiotic. Meanwhile, if we look at the biofilm growth, we can see that there was no biofilm quantified for the oxylipin treated, which now confirms that oxylipins 1 and 4 have a selectivity index, or it selectively inhibits the biofilm and not the planktonic cells. So we also want to know if our compounds are effective against preformed or mature biofilm. So take note, this is for the already established biofilm. So we use two assays for this. So the first one is the metabolic inhibition presented by the bar graph, and the second one measures the dispersive activity of our synthetic compounds, which are indicated by the line graph. So to understand better, let us dissect the graph. So first, for the dispersive activity, we can see here that the enzymatic activity is shown by the right y-axis. And based on this graph, even though that we increase the concentration of our oxylipins 1 and 4, we cannot attain a good dispersive activity. So hence, we can conclude that both oxylipins are actually incapable of disrupting mature biofilm. For the metabolic inhibition, as shown by the left y-axis, we can see a 30 to 60% metabolic inhibition against the preformed biofilm by applying oxylipins at concentration 16 to 128 microgram per ml. So based on these observations, we further evaluated other physiological targets of oxylipins, and from these micrographs, we can also observe that there is a reduction of EPS. So EPS here is these thread-like structures, and it is important that we must take note that EPS is important adhesion molecules which bind together our planktonic cells. So without them, it is possible that your cells may remain on the planktonic form. So aside from the EPS absence, cell aggregation, which is typical to the negative control, is also absent in the oxylipin-treated slides. Moreover, we also observed significant downregulation of staphyloxanthin or pigment production in staph aureus. So to validate. So those are morphologies. So we want to know what could be the regulation in the gene or the protein level. So to validate the observed morphology, for example, the EPS, we utilized gene expression assays using the ICA operon. So here we quantified two genes, the ICA-R, which serves as the repressor, and the ICA-B, which deacetylates or strengthens your EPS. And based on this figure, we can observe significant upregulation of the ICA-R or the repressor, which correlates to the observed downregulation of your ICA-B. So now that we have established the connection between the phenotype and the gene expression, we can now probe for possible protein interaction which happens upon the supplementation of the oxylipins. So for example, we can eliminate here PSMs and proteases because as mentioned before, we did not see dispersive activity of the oxylipins. But we can now focus on the AGR-SRA systems which are correlated to the observed morphologies a while ago. So using Autodoc and Chimera, we were able to visualize the protein interaction of SARA between the ligands tested, which includes oxylipins 1, 4, and 5. So based on this docking experiment, hydrogen bonding were formed and can be traced from the epoxide moiety and the carboxylic acid and the terminal carboxylic acids, and negative binding energies uh, were detected, which validates the favorable interactions of our protein ligand complexes. And this was also same observations was noted from the AGR proteins, the formation of hydrogen bonding, negative binding energies, and the conserved hydrophobic site. So as a conclusion, overall, the conclusion can be presented by this figure. So overall, if we are to rank the analogs that we tested, the highest selectivity can be observed from the trans epoxide bearing fatty acids, followed by the cis isomer, and then the unsaturated oxalipins, and the least or non-effective saturated fatty acids. And then we also observed selective inhibition towards gram-positive bacteria. And in terms of temporal targeting, it targets the early to mid stages of your biofilm life cycle, which falls under the transition of from planktonic cells to the mature phase. So how, specifically how, it targets EPS production and interferes with column sensing signaling. So this study highlights the interconnectivity of the genes, proteins, and more to the morphotypes of Staphylococcus. So by synergizing traditional microbiological tools with gene expression studies and more conventional docking experiments, we can now utilize or we can understand further the mechanistic strategies of natural product-inspired compounds such as these modified oxylipins. And that would be all. Thank you.
So right on target. And now we'll move on to the new methods for the chemistry and biological activity analysis of marine natural products. So the first speaker is Sabrina Bernice Malto, graduate of MS Chemistry, and she will talk about linking genomes and metabolomes of marine microbes for natural products production. So
Asan yung presentation?
Ito po.
Ito. At tayo matulog. Ha? Yan pa ba? Bakit hindi siya nakapresentasyon? Ano nga?
Ayan po.
Yan.
Okay na. Okay na, okay. Okay.
Yan.
Good afternoon, everyone. I'm Zab. I'm going to present to you some of the methods or contemporary omics methods done in our lab for us to aid us in marine natural products discovery. So my talk is entitled Linking Genomes and Metabolomes of Marine Microbes for Natural Products Discovery. So to start, we know that advances in genome sequencing have enabled the identification of far more biosynthetic gene clusters as compared to what was originally purified in the lab. So this suggests the presence of what we call as silent or cryptic biosynthetic gene clusters. And at the same time, advances in analytical instrumentation, particularly hyphenated techniques such as combining chromatography and mass spectrometry, have made possible the simultaneous analysis of the total metabolites produced by our organisms, or what we call as metabolomes. So to keep up with the wealth of data, several tools have now been developed for us to mine genes and secondary metabolites. So since genomics and metabolomics, as what we heard earlier from Ma'am Lil, are the typical strategies employed to determine the biosynthetic potential of your marine organisms, integrating datasets are now performed to accelerate the identification of metabolites and link them to their biosynthetic pathways. So for genomics analysis, from the genomic DNA sequences, we can analyze the production of secondary metabolites by looking into their biosynthetic gene clusters. So tools such as Anti-SMASH and PRISM compare DNA sequences to all known gene clusters compiled in their databases, allowing for the prediction of chemical structures and identification of compounds produced by our organism. So what's also good about these tools are that they are free and have options for web-based analysis. For metabolomics, the preferred method would be mass spectrometry-based metabolomics. So mass spectrometry utilizes small amounts of extracts and can be linked to chromatography techniques. The most popular method for metabolomics analysis would probably be molecular networking, which is our favorite tool in the lab for natural products research. So this molecular networking was made publicly available by through the GNPS platform, which was developed in 2012. It is a web-based platform. It contains many metabolomics tools in one ecosystem, and it has a robust database containing experimental MSMS data where you can compare your own generated data. So currently, the library has more than 360,000 compounds deposited to date. So how does molecular networking work? So in the case of genome mining, we compare DNA sequences. But for molecular networking, we compare MSMS fragmentation data. So it is based on the premise that for a mixture of compounds, those that have similar chemical scaffolds will fragment similarly. And these fragments are detected by your mass spec. So these masses are then compared to the fragments of other molecules in your sample, and if the peaks match, they will form a cluster. So each node represents an MSMS spectrum of a compound, and here the nodes are denoted based on the charge mass of your compound, and the edges or the lines connecting each node are denoted by the similarity score or the quotient score, or sometimes you can put in the mass differences. So
As we can see here, for this mixture of compounds, these compounds cluster together because of the presence of the tryptophan moiety, while this group of compounds formed a different cluster because of the presence of the imidazole moiety group here. So, today, I will present two case studies of two organisms which have different features in which we utilize these tools for secondary metabolite discovery. So the first one is a sponge-associated Streptomyces species. We call it as a canonical example because we can readily sequence its genome and link it to its metabolome. So the second case study is on the harmful algal being causative dinoflagellate gumbo discus species, which we call as a non-canonical example because we don't have genomic data. So how do we use these methods to look at their biosynthetic potential? So the first story is on the sponge-associated microorganism. So Bernard, who was an undergraduate student of the lab, and Kuya Kevin, who was an RA before, were able to purify and recycle pentapeptides from this microorganism. So the structures were assigned using NMR, so we wanted to know if the microbe can still produce other compounds. So we performed whole genome sequencing, and we found that its best 16S match was this strain of Streptomyces cacao with 98.6 sequence similarity. So analysis of the whole genome sequence of this organism suggested that a significant proportion of it was dedicated for secondary metabolite production. And most of the secondary metabolites, or most of the BGCs, are encoding for non-ribosomal peptides, or NRPS. Since we know that the BE pentapeptides are produced by 843A, we analyzed the whole genome to find the BGC coding for these cyclic pentapeptides. So we found two NRPS genes encoded in tandem, both with 5 adenylation domains, so meaning they are 5 amino acid peptide products. So a paper by Roman Hurtado in 2021 suggested or showed that for this strain of Streptomyces cacao, the first NRPS is coding for the BE cyclic pentapeptides, while the second NRPS codes for the pentaminomycines. The main difference between the BE pentapeptides and pentaminomycines is this group of glutamic acid here, which is converted into an arginine moiety. And the cytochrome P450, which is found in the BGC, oxidizes the arginine moiety into an 5-hydroxyarginine in pentaminomycines. So we then analyzed the crude extract using GMPS molecular networking, and we're able to generate this molecular network containing 9 clusters and 69 lone nodes. This node, which was annotated by the GMPS spectral library as B18257E, showed good matching with the reference spectra. And although the analog B18257B was not reflected in the molecular network, a peak that corresponds to this analog was detected in the LCMS profile. So fragmentation analysis of this putative BE peptides confirmed the peptide sequence. Since the genome also suggested the production of pentaminomycines, we examined the presence of this class of peptides in the extract of 843A. So manual inspection of the other nodes in the molecular network was performed, and we found this cluster of compounds or nodes that correspond to the masses of pentaminomycines A to D. And aside from that, we were able to identify a node that clustered with the pentaminomycines, which has a mass that has not yet been reported in literature. So using fragmentation analysis, we suggested the planar structure for this putative pentaminomycin analog as shown here. And through metabolomics, we showed more analogs for pentaminomycins, which was not apparent in the BGC information. And with the data from the BE pentapeptides and the pentaminomycins alone, we can already show that the genome data corroborates with the metabolome, in which we saw predominantly peptide-based molecules of the NRPS type. So another class of peptides that we observed in the genome of 843A are RIPs, which another kind of peptides that are ribosomally synthesized. So we're able to map the cacao bean biosynthetic gene cluster at 99% similarity. So cacao bean is a lantipeptide, which has this lanthionine moiety here, which is synthesized by the lantipeptide synthetases, which is found in the biosynthetic gene cluster. So although our sequence or BGC detected for 843A is truncated at this region as compared to the reference BGC, we can see that R3843A still produces this peptide because of the presence of glycosyltransferases, which adds this sugar moiety here, as well as the 100% sequence similarity of the precursor peptide. And to probe the production of cacao bean in our metabolome, we performed metabolomics analysis as well. So manual inspection of the MS1 data of the crude extract showed masses corresponding to the triply charged ion right here. And naturally here are some of the doubly charged ions suggesting the production of cacao bean, but at very low amounts. This also corroborates with the difference in the regulatory genes, this one here in green, which we're not able to identify in 843A. So for 843A, we assigned 7 NRPS genes or peptides and 1 linked peptide from the genomics data using MS-based metabolomics. So how about for microbes where the biosynthetic gene clusters are difficult to mine? So not all genomes can be analyzed as easily, such as in Streptomyces species. In case of dinoflagellates, they are known to have enormous genome sizes and permanently condensed chromosomes. So here we applied metabolomics for the emerging hab-causative dinoflagellate Gamberdiscus species. So we know that Gamberdiscus carpenteri was isolated from Bolinao for the first time and reported in 2018 under the lab of Mamrod, and it showed CTX activity through RBA or radioligand binding assay. And there are also Gamberdiscus balekei species that were isolated in Bolinao and Samar in 2017 and were reported by Dr. Gary Benico. So to probe aside from the CTX RBA data that was reported in Gamberdiscus carpenteri, there's very limited chemical information and toxicity information on this Gamberdiscus strains isolated from the Philippines. So to probe this, we have collaborated with BIFAR for toxicity studies, and we performed MS-based metabolomics and molecular networking techniques to assess the biosynthetic potential of these hab-causative organisms. So we cultured the 3 Gamberdiscus strains and subjected them to chemical extraction, refractionation, and LC-MS analysis. So we also analyzed an authentic standard of PCTX3C, which is a ciguatera fish poisoning toxin. So we analyzed this one in combination with the extracts for us to tease out if a compound from our extract will cluster to PCTX3C. So using molecular networking, we generated the molecular network of these 3 Gamberdiscus strains as well as the PCTX3C standard. So the entire molecular network consisted of 45 clusters and 271 single nodes, and we found that the seed compound PCTX3C appeared in the molecular network as a single node. So this indicates that the metabolites present in the extracts of Gamberdiscus strains did not show any similar fragmentation pattern as PCTX3C. So looking into the other nodes in the molecular network, we observed this cluster with a mass of 1021 daltons in the 3 strains. So the replication of this mass suggested that it corresponds to the pseudomolecular ion of a waterless peak of 4,4 methyl gamberone, which
is also a polyether ladder frame toxin that is different to ciguatoxins. We then confirmed the presence of 44-methylgamberone using manual fragmentation analysis using MSMS and MS data. So here, this includes the molecular ion peak, a series of water losses, a sulfite loss followed by a series of water losses, the breaking down of the I ring as well as the removal of this alkyl chain here. So the presence of 44-methylgamberone is also supported by the observed ciguatoxic activity through the radioligand receptor binding assay. So this was done by our collaborators in Japan. And shown here is the result of CTX-RBA of the crude extracts of Gamber discus in picogram brevetoxin equivalents per cell. So G. carpenteri gave the highest toxicity, while the G. balekii strains from Samar and Bolinang showed comparable toxicity. So what about the other compounds that are present in the metabolome of the strains? So using GNPS, we're able to annotate fluorophoribide, which is a chlorophyll derivative, pyridinine, which is responsible to the orange to brown appearance of Gamber discus cells, and a strain-specific cluster of polyhydroxylated compounds is also identified that's specific for Gamber discus carpenteri, as well as a peptide cluster in which is specific from the G. balekii strains from Samar. So this may provide chemical evidence to the NRPS PKS hybrid sequences identified in the transcriptome of some Gamber discus strains. So what is also evident is the convergence of nodes from both Gamber discus balekii strains, which are highlighted here in orange. These species and strain-specific strains or clusters may be potential chemotaxonomic markers for the identification of particular Gamber discus isolates. However, GNPS was insufficient in the replicating compounds from these clusters. So this highlights the limited information on the metabolome of Gamber discus and other dinoflagellates. And further studies towards identifying these compounds are needed to know the potential utility of these compounds as chemotaxonomic markers. So in summary, using canonical genome and metabolome integration, we were able to assess the chemistry of Streptomyces cacao. So the data we presented provides potential avenues to purify new relatives of known molecules and clusters not replicated in GNPS, as well as to utilize the metabolomic information as a template to prepare new analogs using organic synthesis. And the metabolomic information. So as for the non-canonical analysis of GNP of Gamber discus species, we're able to demonstrate the chemical richness of the secondary metabolites using GNPS, using molecular networking, and additionally, this method allowed us to visualize secondary metabolites that are strain or species specific, which can be potential chemotaxonomic markers. So we'd like to thank these funding agencies, specifically DUST-PCHRD for funding the 843A study, and DUST Picard under the haphazard program, the Red Tide Lab and Biome Lab for the cultures, and the secondary metabolites profiling lab of University of the Philippines, Diliman.
Okay, and for the last speaker, we have Zildjian Akyatan, graduate of MS MBB, and she will talk about no pain, no gain, teasing out pain signals for neuroactive compound discovery.
Dito ako. Para sa'yo.
Kailangan bang pointer?
Yes, ma'am.
Pointer?
Okay na?
Ayan. Ito yung pointer.
Gumagana yung next one.
Yung next, sabi ko sa'yo.
Ayoko dun. Dito, tatayo.
Ano ba yun?
natin lang.
Ayan na. Move forward. Move forward. Pointer at the back.
Pointer.
Move forward.
Okay. Ayun. Okay. Hi everyone. So I'm the last speaker. Okay pa ba kayo? Kaya pa? Okay. So my topic is about the method that I've been using and has served me well and served the lab well for the past X years. So it's about the DRG assay. So show of hands. Show of hands to the people here. Some 20-something RAs who's already having back pain, shoulder pain, knee pain, hand pain. Wala show of hands. Yung mga umalis kasi they have field, so carrying big crates, sa scuba diving, rolling back. So all of those pain, including the pain that we are feeling right now with intense heat, di natin nararamdaman but with intense cold. Oo, ngayon siguro. Also, somato sensations like proprioception, balance, touch. These are all felt in the neurons in our body. But actually, these are transmitted from the PNS to the CNS by ganglions, which are called DRGs. So what are DRGs? Where are they and where are they found in the body? So these DRGs are found in the spine, and they are these guys, so the bulbous. So in humans, like what I said, the circuitry, the electrical impulses are felt sa mga periphery natin, and then the electrical impulses goes through the body into these DRG neurons, and then from PNS, it sends it to the CNS. So iniipon, those sensations are pulled, collected in the CNS here, in the PNS here in the dorsal root ganglion. So why? These guys, these DRG neurons, are actually filled with different cells, a heterogeneous population of cells that have a variety of receptors, ion channels that can be receptive to those types of pain and touch and feelings that I've mentioned a while ago. So in the assay, this is what we want to take advantage of, that heterogeneity. So in MSI, in our lab, we use mice for obvious reasons. Because bawal sa tao. Within mice, we use 3 to 4 week old mice. We get their spine, we dissect the DRGs. So I'll show here a video of what we usually do. So this is the spinal column of a 3 to 4 week old mice. So we cut it off. Sorry, medyo gruesome, but this is the spinal column, and in each vertebra there is a pair of DRG. So makikita in here, we have here, here, and here. So in mice, we get it from the L1 to L6 because the lumbar is what innervates most of our somatic sensations. So sa kamay and sa paa. So we get approximately 6 pairs of DRG in every mice. So yan, makuha siya dito. So we get all those 6 DRG. So they're like bulbous structures. So they're like pomegranate. So when you open the pomegranate, you have a lot of seeds, diba? But in DRGs, those seeds are actually different cells. Each one is different from one another. So we get the cells, we culture it. So the DRGs are actually those bulbous looking structures. We clean up the structure and then we plate it into a 24-well plate. So these are primary cultured neurons. Here, so this is the final product. Now, you may ask, bakit ka nalang gumamit ng mga immortalized cell lines? Well, what we really want with the DRGs are its heterogeneity of the population. So these primary cultured neurons can only be used 24 hours. Because if you go beyond 24 hours, they'll already differentiate into homogeneous type of cell. And what we want are the heterogeneous population. We just want to test the samples that we have in those receptors while they still have it in their surfaces. So what's nice about the platform is in the assay, we can actually individually select each type of cell and monitor their phenotypic changes. In the assay, we use a calcium imaging assay, calcium imaging platform, using a Ph.D dye. So the dye, essentially, we load, so this is an individual DRG neuron, for example. We load the dye, we load the cell with the Ph.D dye, and this Ph.D dye is ratiometric, meaning it has 2 conformation and it has 2 signals. So you have a signal at 380, meaning a type of dye that hasn't been sequestered yet, or wala siyang calcium, and you have the signal at 340, which has dye bound with calcium. So at the start of the assay, we put an excess amount of calcium in the surface of the cell, but this is in a buffered condition, so the calcium remains outside. But when we use KCl, we change the resting membrane potential. We perturb the cell. So you cause a depolarization that allows the calcium to go inside the cell. So with the X amount of 2AM inside, you will now see a shift from this guy to this guy. So you're gonna have an increased signal in 340 and a decreased signal in 380. That's why when we remove the calcium, you will revert it back. You'll have more of the 380 and then less of the 340. This will now show us the 340 and the 380 will essentially give us these kinds of peaks. Now these peaks will denote how calcium goes inside different VGCCs, GPCRs, or voltage-gated calcium channels and deep protein coupled receptors that is on the surface of each cell. And each cell will now have a different combination of these guys, and this means that each individual cell will have this kind of trace. Now, how do we use it in our assay? So like what Ma'am Lil showed a while ago, we will have two baseline pulses. And with the sample, may it be from a crude extract from microbial extracts, phenome extract, or something else, we can see its effect on this succeeding KCl pulse. Essentially, each pulse will give us a real-time measure of calcium, and that's what we're detecting in this assay. And usually, a sample will have several activity if it's active. It could have a direct effect wherein the moment you add the sample, it will already cause an increase, change in an opening of, let's say, an ion channels that will allow the increase. That's why we can see here direct influx. You can have indirect effects wherein after the incubation, we can either see an amplification or a block or an amplification. And then you have an inactive cell. So, sabi niyo bago, but actually this assay has been inside MSI for quite a while. So we got this assay from the lab of Dr. Balomero Olvera with postdocs Russ Deckard and Alan Light in the University of Utah, and then they transferred the technology, the system, the platform here in the Philippines in the early 2010s. Ganyan, 2010 sila, and then ako mga 2017. And then now, these are projects from Dr. Giselle Concepcion, and now we have it in the lab of Dr. Lilibeth Salvador-Reyes. So in the MSI, we have optimized, troubleshoot, and have the assay now as it is because of these projects, the DHP Pharmacies Phase 1, PMSI CBG, and the DHP Phase 2. So the title of my talk is Teasing Out Pain Signals for Neuroactive Compound Discovery. So where can you use the assay? Do you just use it in parts of the pipeline? Do you use it in the start, the screening? Do you use it in target identification? Now the answer is we can use it everywhere. We can use it top to bottom, bottom top. So in the screening, I have an example of what we did in the PMSI CBG, where we already screened 1,000 plus crude extracts and found approximately 3.4% of hits. Now, those hits can be true hits or nuisance hits. And we use the help of mass spectrometry, the replication, to remove the nuisance hits. So I'll give you an example of nuisance compounds that are also active in the assay. So these are beta-harbolin compounds, harmine, indole, hapalindole, that will, so nuisance compounds are compounds that are well known. Their bioactivity is well known. So if our goal, our motivation is to look for novel compounds, then we want to shy away from these guys. So if these guys are already active, then it will lead you to a different path. So thanks to mass spectrometry, we can already replicate it. So in the purification, we can avoid these guys. So with the help of DRG screening, mass spectrometry, we can weed out nuisance compounds so that we can push the discovery to novel compounds. Where else? Where else can we use DRG assay? You can use it in bioactivity guided purification. And this means we can use the assay about the purification. So I'm showing here an example of crude extract that when we did purification in the fractions, we saw two distinct activities. We found one activity in fraction 1 and one activity in fraction 6. Now, we use the DRG assay to do the fractionation, follow the activity, and now we can actually connect it to the pure compounds that causes those activities. So this tells us that the DRG assay is quite replicable, consistent. So as long as the peptide or the compounds are stable. So it tells us that you can do repetitions of the assay, and if using the same compounds, you will get maybe a crude extract with that active compound. It will have the same phenotype, so you can follow it down to its purification. Target identification. Now I'm going to show you an example of how the DRG assay was used to narrow down the target. So here we have pharmacological differentiators of trip channels. Trip channels, we have AITC, which is the main ingredients in wasabi. So allyl isothiocyanate. So we have here a neuron that has a signal for AITC but doesn't have a signal for menthol or N-capsaicin. Therefore, this particular neuron has trip A1 because trip A1 is the target of AITC or the wasabi. For menthol, I have here sample ng salon pass, we have a neuron that has menthol trace or it has responded to menthol, but no AITC and capsaicin. And lastly, we have a trace here that shows you capsaicin trace, but no AITC and menthol. So this differentiates three different neurons, one that has strip A1, the second that has strip M8, and the third that has strip V1. So that's the power of the DRG assay. And in tandem with cell size analysis and RNA sequencing, we can narrow down the target to a specific subtype and maybe into a specific type of receptor. Lastly, for mechanistic studies. So here we use modulators, agonists, and antagonists to make sense of the pharmacology. And how do we do this? So I'm going to show you an example of how we determined a particular subtype of neurons in the DRG assay that has the alpha-7. So acetylcholine here is a neurotransmitter, but it opens up nicotinamide acetylcholine receptor, specifically the alpha-6, alpha-3, beta-4 subtype. Now we use PNU, which is a specific modulator of alpha-7. So here we have three different subtypes that we want to play around with. Now with PNU, we don't have any signal. You have acetylcholine, you have signal, PNU, no signal. But when you put acetylcholine and PNU together, you have an increased signal. Therefore, this is saying that this particular neuron here has both the alpha-6, alpha-3, beta-4, and that the increase that we see here is caused by the alpha-7 subtype. So those are the kinds of questions we can ask. We can also do this with calcium channels. We can also do this with NAV channels. And later on, I will show you what we did with one of the peptide we did for sodium channels. All right, so the DRG assay can be used in all four of this part. And when you just ask the right question, then maybe you can have a star compound. You did it. So today I'm just gonna give a brief highlight on two of the star compounds we've identified using the DRG assay. So one from a conus sample, conus tessellatus, and another from a cyanobacteria from cyanobacteria. So here, what I'm showing you is what the genome of a tessellatus looks like. So it has a lot of peaks, meaning it has a lot of toxins. Usually in our pipeline, what we usually do is test it just with ICMBA or intracranial mouse bioassay. But what can you see here? In the 7 pools of fraction, only 2 is active in ICMBA. Now, if we only follow ICMBA, then we're already neglecting 4 fractions worth of peptides. And in this particular thesis by Ate Chai, she was able to identify 2 peptides from those guys that are not active in ICMBA. So this tells us that the DRG assay is covering a wider platform, wider lens to look at. So that's the story for cetus stellatus. Essentially, two new peptides, which has until now, it's still an orphan peptide. We're still looking for answers on its specific target, but you know, we're trying and asking the questions to answer this. The next star compound that hasn't been published is another compound isolated by Kuya Gemay. So in the screening, we saw that this particular compound blocks the calcium influx in the DRG assay. And it blocks 55% of the cells, so maybe half and half. Now, what they found out after Kuya Gemay did the structure identification, they saw that this compound was structurally related to a sodium blocker. So now we ask the question, is this really a sodium channel blocker? And we did that with constellation pharmacology using agonist and antagonist of the sodium channel. So we use veratridine, which is a sodium channel agonist. So it opens up sodium channel, and tetrodotoxin, which is from pufferfish, which is a known sodium channel blocker. So here we have two types of cell in the cell population of the DRG. We have a veratridine sensitive and veratridine sensitive and veratridine insensitive. And we see that with this trace. So we see a K, which should open up the calcium, and we have a K and V, which opens up both the sodium and the calcium. So you know that this guy over here has the sodium channel that is open, meaning it contains sodium channels. And this one, maybe it contains, but it's not specific to veratridine. Now we use a known blocker of the sodium channel to see if that hypothesis is true. And what we can see here is that that increase by veratridine caused by the open up of sodium channels is blocked by TTX. And in this type of neuron, since there's no sodium channel to block, you don't see a decrease in its height. So this neuron here, or set of population here, is TTX sensitive, while these are TTX insensitive. And so we add Kuya Gemmay's compound 1. Will it act like TTX, or will it block cells that are also TTX insensitive and veratridine insensitive? And what we see here is that compound 1 has the same phenotype as TTX, and it also blocks the same neuronal population as TTX, saying that, yeah, maybe that this compound really is a sodium channel blocker. So you can see it here. This is the neuronal population. We have the veratridine sensitive, or the red ones, the blue ones are insensitive, and that 1/3 of these guys are blocked by TTX, and that 100% are blocked by compound 1 and TTX, suggesting that our hypothesis is going towards the right direction, that maybe truly compound 1 is a sodium channel blocker. So this just tells us about the power of DRG assay. It's a method-based discovery of neuroactive compounds and holds a wide selection of targets we can test simultaneously. So instead of testing it in individual cell types, individual cell lines, or overly expressed, let's say, NAV channels in a HEC293, you're actually testing a wide variety, a wide selection of receptor channels in one go. This gives us a higher rate, at least in our lab, higher rate of finding new compounds. Like what I mentioned before, it's robust and replicable, and we can functionally validate it with agonists and antagonists. Future directions, we can use advanced subtype differentiators. We can tandem it with RNA-seq to narrow down targets even more with samples. And since right now we're using 200 to 400 cells, with the automation, we can increase that power to 1,000 cells. So to end the talk, this is the DRG. This is the power of the DRG. It's one cell contains a lot of receptors already. And it's not just one cell. You have other DRG cells that contains a lot more receptors. So you're essentially testing a set of sample in a number of receptor, meaning you're already saving up material, saving up time, while also testing probably orphan compounds or orphan receptors. And with the use of agonists and antagonists, we can narrow down the target. And with narrowing the target, now we can use through the lens of pharmacology to identify which among those receptors are the true target of the samples or the compounds that we're able to isolate. So I end this talk by going back to my title, which is No Pain, No Gain, teasing out pain signals for neuroactive compound discovery. We tease out pain signals, and these pain signals are like puzzle pieces that we have to decode using pharmacological techniques. And the no pain, no gain is true to both the assay and our because it's so much easy to showcase an assay with success stories. But actually the assay we did not see in my report or in my talk are the failures, the experiments or where we asked a lot of questions that brought us to more questions than answers. Um, with that, I'd like to thank these people, the old guys, the new guys, and the upcoming guys. And no pain, no gain. Like in life, it's no pain and no gain. So, ano sabihin ko, ma'am? Meron ako ano skill eh. Ayun. But it is what it is. Life is no pain, no gain. It's about what matters most is the journey. The journey. And more importantly, the people we meet along the way. So with the growing pains, the learning pains, and the back pains, happy 10th anniversary to MPL, to more science, more questions, and hopefully more answers. Thank you.
No pain, no gain. But no pain, no glory. We have a batting rate of 95% graduation rate, at least one publication for every graduate student, and attending one international conference or one international training for each student. Okay, floor is now open for questions for our four speakers. Any questions? How about in Zoom? Are there any questions in Zoom? Maybe Zildjian, you can elaborate on the how many replicates you do for DRG in terms of biological replicates and statistical replicate, because of course reproducibility, rigor, that's very important for your experiments to be able to differentiate real versus response.
So since we're working in a lab where we try to purify things, the crude extract to the fraction to the pure compound is already a biological replicate. And in the assay, we can also devise a technical replicate in one well, we can test the same compound in just one in one experimental design. We can test the same compound, and if it targets the same cells and it has the same phenotype as the same cells, then that would be a technical replicate. But to answer Mom's question, we use 2 to 3 biological replicates and then at least 3 technical replicates.
Okay, any other questions? Wala nang tanong? Mia.
Hi, question for Kuya Jeremiah, dun sa, sa structure elucidation. I wanted to know, like, what is the scale of, like, how much did you have to culture for the cyanobacteria and what is the yield? Kasi parang ang daming processes that involve chemical derivation. That means you are losing material. So you need a lot of starting material. So I just wanted to know what is the scale of the culture and the yield for that particular compound. Ayun, si Wormicyclamide. So I think ang tagal natin mo 2015, 2016.
So I think for this I think the first culture it gave me around 0.5 milligrams for a 2-liter culture. So yeah, that's not too bad for cyanobacteria. And then we did another extraction, about 4 batches of extraction. So I think that's enough. The 4 batches of culture, I think 2 liters times 4, 8 liters, and it's enough for the whole structural oxidation, including the absolute configuration. Well, because compared to other microorganisms, marine microorganisms, for example, yung thesis ko, Teridinibactin is actually cultured 86 liters to get 10 milligrams of Teridinibactin A, but only 0.3 milligrams for bromoteridinibactin A, and then 0.1 sa other new analogs pa. So it's really not too bad for cyanobacteria compared to Teridinibacter ternary.
So actually for the maybe to add to that, for the structure elucidation, no? Determining the planar structure, you're actually essentially using very little material. Your NMR, you can recover back your material. Mass spec, you only use a few microgram, very little. With the total structure elucidation, using derivatization and all that, we did not use a lot because we moved to a mass spec based method. So you can derivatize with actually 100 microgram of material, do an MRM, which is very specific for your target analyte. So you dilute out all the noise, and your target amino acid for the chiral analysis will actually have a higher signal. So it's a combination of an improved methodology and at the same time using different platforms or separation techniques, because one part is separation technique, which is your chirobiotic tag and the FDLA. But if your detection method, the other part is a detection method. If you use an HPLC UV method, you will need to use a lot of material. But with mass spec based method using MRM detection, you can get away with 100 micrograms. And that really certainly helps in stretching your material. Okay? Any other questions? So, no pain, no gain. You need to culture a lot. That's the reality of it all. Natural products, it's not, in all honesty, it's not very easy, right? Because you're dependent on your starting material for all your downstream analysis. It's really hard work. And if you fail in one step, you'll have to repeat again from the beginning, because that's your material to the next level. And like for synthesis, you get your starting material from the shelf, conjugate it again, that's it. But natural products, you have to start from culturing. It requires some patience, but we all get that, boss and students alike. So with that, I'd like to thank everyone for attending this afternoon's presentation. I hope you guys learned something. We appreciate all our collaborators and the other students from other groups whom we worked with in the last few years. So thank you very much, everyone. For those who are here at the MSI AVR, I think there's some refreshments outside. And if you want to drop by the lab, we also have a small exhibit showing how we do the collection of chronos, and at the same time, microbial purification. We prepared it for the MS1 lab tour last week, but certainly everyone's invited if you want to see in real life our workflow pipeline. So thank you very much.